Uh, Good morning, everybody. I, Devesh, a member of JAX Foundation, welcomes you all at the National Summit 2020 Innovation in Biotechnology Era. I would like to introduce our keynote speaker of the day, Professor Dr. Shilpi Sharma, Professor Savitribai Phule, Pune University. Dr. Shilpi Sharma has completed her graduation from Institute of Home Economics with Bachelor's of Science (BSc) in Microbiology in 1999 and did her post-graduation from Dr. B.R. Ambedkar Center for Biomedical Research, University of Delhi with MSc in Biomedical Sciences in 2001. She did her PhD in molecular immunogenetics from Institute of Genomics and Integrative Biology in 2006. She is currently working as a professor in Savitribai Phule, uh, Pune University. Now I take the privilege to invite her for sharing her thoughtful and fruitful words. Good morning, ma'am.
Good morning. Thank you for those kind words. Very good morning to everyone. It's an absolute honor for me to be here today and to welcome our Honorable Minister, all dignitaries, participants, and delegates of the National Summit 2020 Innovation in the Biotechnology Era. This summit has been inspired by the idea of the United Nations Programme of Sustainable Developmental Goals 2030 that emphasizes upon the idea of continuous contributions and innovations. Other than that, it is also inspired by our Honorable Prime Minister, Sri Narendra Modi's 8-point vision for the 20th century for India, with the idea of uniting the entire country by adopting good governance and the use of technology to edge forward. The National Summit 2020 would help us on a massive scale, particularly the young minds, in several ways. The summit would provide an opportunity for research interaction, which indeed is a privilege for the young researchers to learn and expand their knowledge. It would also work as a platform where biotechnology professionals, academicians, practitioners, and government officials will be able to share their ideas pertaining to the applications of biotechnology research and innovation in particular. The JAX Foundation, just to introduce you to that, is an international organization supported by the BRICS International Forum and has been recognized by the Government of India under the Indian Trust Act 1882. Over the years, it has been working for the society, particularly for the welfare of young generation and promoting social or political change. It has indeed played a very important role in developing the society, improving communities, and promoting citizen participation, particularly for the youngsters who are able to fulfill their dreams here. JAX Foundation this year has joined hands with Symbiosis School of Biological Sciences. This is an endeavor for the development, representation, and advancement of different areas of biotechnology. The aim of this association is to promote, advance, and disseminate the interest of scientifically oriented biotechnology research and application for the benefit of an individual and the society as a whole. This national summit will provide the platform for scholarly exchange of research ideas in the different areas of biotechnology disciplines. We all know that COVID-19 has taken an uncertain toll and has affected so many people all across the world and has made it to stay within the four walls. It would rather not be advisable to affect today's summit amidst this pandemic, and we are all waiting for the COVID-19 vaccine, which is again an application of biotechnology. We are very lucky to have internet service as a luxury source to propagate our ideas, regulate the awareness programs, share opinions, and help one another after even being so apart from each other. I would like to encourage all delegates to participate with full enthusiasm and wish everyone a successful, safe, and fruitful summit participation. The chief guest for today is our Honorable Minister, Sri Satyendra Kumar Jain. Sri Jain is a cabinet minister in the government of Delhi and is a politician from the Aam Aadmi Party. He has 7 cabinet ministers in the government. He is one of the 7 cabinet ministers in the government of Delhi, led by Mr. Arvind Kejriwal. His portfolio includes health, industries, home, public work, department, and power, urban development, and transport. However, due to some prior commitments, he has been unable to come over and has sent his greetings in the form of a video message. I would now like to request Ms. Tanya Bhagat to play the video. Tanya?
नेशनल बायोटेक समिट 2020 2020 में भाग ले रहे सभी छात्रों को मेरी ओर से बहुत बहुत हार्दिक हार्दिक शुभकामनाएं। बायोटेक्नोलॉजी विज्ञान की वह शाखा है जो हमारे देश और दुनिया का भविष्य तय करेगी। आज इस समिट में हिस्सा ले रहे छात्र कल के वैज्ञानिक बनेंगे और विश्व में एक क्रांतिकारी बदलाव का साधन बनेंगे। मैं एक बार फिर से आप सभी को बहुत बहुत बधाई और शुभकामनाएं देता हूं। आपका यह आयोजन सफल हो।
Thank you, sir, for those kind words. I really appreciate you, sir, for taking out your time from your really busy schedule. It has always been a pleasure to have your blessings with us. Now, I would request Ms. Khyati to take this forward, and I wish all the best to all the participants and delegates. Thank you so much.
Now we will start with today's plenary sessions. I would like to introduce our today's moderator for the session, Mr. Aditya Acharya. He is an executive member of JAX Foundation. Currently, he is also a member of the Global Microbial Identifier Group, Singapore, active member of Federation of Asian Biotechnology Association and Indian Microbiology Society. He is currently doing research in the field of computational drug designing to find lead candidate for SARS-CoV-2 virus. Now I would like to call Mr. Aditya Acharya to take this event forward. Over to you, sir.
It's my great honor, first of all, to be invited here for the second day as a moderator. We are living in an era of new ideas, creativity, and innovation, where all three aspects are contributing greatly towards the development of our society. So today, at a place where both the process, a new product, and also shaping of a new product, where biotechnology is immensely related with our lives. Standing today in this national summit, the theme for which is aptly described, which is innovation in biotechnology era, the need of the hour. So now I would like to call our very first speaker of this very great morning today is Dr. Lopamundra Das Roy, founder and AMP president, Breast Cancer Hub. Dr. Das Roy did her postdoctoral fellowship from Mayo Clinic, College of Medicine, USA, in cancer immunology and therapeutics, focusing on breast and pancreatic cancer. She received her MBA from Northwestern University, Kellogg School of Management, Chicago. She founded the Breast Cancer Hub as pro bono and voluntary service with a dream to save lives by making impactful changes and working at the grassroots level. Well, we welcome you, ma'am.
Thank you. Thank you very much. It's my honor to be here with you all, and thank you, Shilpi ma'am, for the welcome speech, and it was really nice. I'm humbled to be here. I need to share my presentation. So can I do that from here? I already sent my presentation earlier.
Actually, if you have, you can share the presentation, ma'am. There is a option.
So how do I do that? So if do I have to? Where do I
see? There is a present now option down to in the Google Meet itself. Just click that. You'll get a dialog of options.
Okay, so entire screen. Okay, give me one.
Present the entire screen, and then just open the presentation that you have, the PPT.
Okay.
It should be visible to us. Let's see.
Are you able to see?
No, we haven't got anything yet.
Okay. So I have my presentation open on my laptop, and then I'm going to the present screen, And I'm
Yeah, present
hitting,
now.
yeah, so I'm hitting entire screen. And do you see my presentation?
You need to select the screen and you click on that and then say present.
Okay. Okay.
Yes, now it's coming.
Okay. So now I have to select it.
Now just open the presentation.
So it's open on my screen. So do you still? Okay, let me just try it out again. Okay, I got it. Let me see if I can. Do you see now? Or I shared my presentation earlier. So will anyone, Tanya, if you can pull it up
Yes,
for
ma'am.
me?
Yes,
That would
ma'am.
be great. Yeah.
Yes, ma'am. Sure. I just present
Yeah,
it.
okay. Tanya is doing it. Okay.
Okay, perfect. So shall I stop presenting mine so that Tanya can see this?
Yes, ma'am. Yes, ma'am. Please.
Yeah, I did that. Sorry about that. I don't know why it went. So Tanya, I stopped presenting, so you can share my screen, please.
Sure, ma'am. Just a second.
Yeah.
I hope it is visible.
Yes, it is. If you could
Yes.
make it full screen, that would be great. Okay, perfect. So, yes, everyone can see my screen and hear me, right? Correct? All right, thank you. If you
Yes,
could
ma'am.
go to the, if you could, thank you. If you could go to the second slide, Tanya, please. To the next slide. Perfect. Okay, so I don't see my graph here. So anyways, that's fine. So this was the statistics that was shown here, but I don't see the graph over here. I don't know why. So maybe we have to download or something. That's okay. So with breast, so my forte is breast cancer. Now, 1 in 8 women can get breast cancer in their lifetime. We all know that. But unfortunately, the death rate is significantly higher in the developing countries, especially India. So I'm focusing my work mostly in India now and also in the US. And what is the main reason behind that? One of the main fundamentals reason, I know we are in a biotechnology forum where we are going to talk about high-end experimentation models and what we have discovered, but something very basic, and Tanya, if you could go to the next slide, please. Something very basic that we forget, which is the taboo. The taboo of using the word breast. And that is the main reason why in India, the detection with breast cancer is so late. And I would just start with a simple story. So I was 12-year old. One of my friends, I was in Guwahati, Assam. One of my friend's mom died. And that was many, many years back. And when I went for the funeral, I asked that what is the reason? Even to mention the word cancer, it was a big hesitation. When I asked cancer where, and then they said breast. So I said breast cancer, I was hushed away. So this is the respect we are paying to a mother that we can't even discuss the topic that cost her life. And it didn't go away. I was in Delhi University. I know someone in Delhi who died of breast cancer, but the family said brain cancer because they have daughters in the family, they will not get married. So irrespective of socio-economic condition, irrespective of education, something basic we forget. And that is the story of Lotika Das, who you see in the middle over here, that she was educated, but when we asked her, my friend's sister-in-law, that what happened, you know what we figured out? She told, I did not know. I did not know that a painless lump could be cancer. So in regard to the taboo, it's not only the taboo, it's the ignorance. Women are not aware. And whose responsibility is it? It is our responsibility to translate this information. Next slide, please, Tanya. Now, one important thing that, next slide, please, Tanya. Yeah, thank you. So one important thing also we forget about breast cancer, that when we think of breast cancer, we think of pink all the time. But men get breast cancer too. Now, though it may be 1 out of 83 men, right now the statistics says that, but that one man can be a father, can be a son, or can be a brother of the family. And the issue that we face with men is most of the research that we have done on the immunotherapy or all the clinical trials that we have taken, that actually always we ran in the females. Be it in the preclinical study, I have always worked with the female mice. We never think of the male mice. The hormones are different, there's so many body molecular mechanisms are going on in a different way, but then when we are treating the male patients, we are treating in the same way. That's why we are still focusing again now, from the clinical aspect, because the clinical trials, whatever, like all the hormone therapy we are giving, we are replicating the same in the male. And the situation with male is worse than female, because male think it's a woman's disease, so they are more shy to talk about it. So from the biotechnology point of view, I would request that if when you are running any experiments on the breast cancer in India, please take into consideration the male transgenic models also, because that is equally important. Next slide, please. Now, in the next transition, what we are going to talk about is what is the screening process, the advancement of technology, right? We know that the standard screening process with breast cancer is mammogram, which is the X-ray of the breast. Now, so I have been intensively working in India. When I ask, like when I come to India, like how many of us over here, if I ask you, how many of you here actually, when we turned 40, did our screening mammogram? Not for diagnostic. We go to the doctors only when we have issues. We should be going for screening. And how many of you here, the male members, actually discuss with your mom, like, mom, did you ever do, did you do any screening for breast exam? That is the request I would want from this panel, the esteemed panel, esteemed students. Please take it forward. It is important. One very important thing that, next slide please, that I'm going to discuss in the next slide is the issues with mammogram. Now, did you know that, like, so mammogram, X-ray of the breast is taken as the gold standard. When we do mammogram, which we are supposed to do after the age of 40 or 45 every year, it comes out to be like, okay, if my mammogram is negative, I should be happy about it. But that's not the case. You can see the picture of Dr. Nancy Capello. She did her mammogram in 2004. Her mammogram was happy mammogram. Now in USA, what happens here is we have a physical every year. Doctors do clinical breast exam. That's again is a taboo in India. Many female doctors told me that, Lopa, you are telling me to examine a breast of the women. They'll be, why are you touching my breast? Nothing is wrong with my breast. That's again a stigma. And the male doctors are equally hesitant. So, but in US, her life was saved at that point because doctors did clinical breast exam. They felt a lump. She did an MRI, magnetic resonance imaging, and she was detected with stage 3C. With breast cancer, the stages are stage 0, stage 1, stage 2, stage 3, stage 4. So from stage 0 to stage 3, we can 100% cure the cancer. But the more the cancer is progressing, the more aggressive the treatment will be. The earlier we detect, is much easier. We can just do mastectomy, and the treatment is also much less aggressive. Now, in Dr. Nancy Capello's case, she was very mad. She asked the doctor that what happened. The doctor said, you have dense breast. The reason I'm talking this in this forum is if you're going through your mammogram, please, you need to learn about this situation that mammograms can also miss tumors, cancers. And why does this happen? When we have X-ray, I don't know if you can see my arrow here. So you see on the most left-hand side, there's a dark, plate here, which means that our breast tissues are mostly supposed to be made up of fatty tissue. When we have fatty tissue, the background is all black. But when we have fibroglandular tissue, which is dense tissue, it appears as white patches, which you can see on the pictures. Why am I talking about white patches here? Because when we have a tumor, a cancer, a lesion, it appears as a white dot. But when we have dense breast, it's like finding a snowflake in the snowstorm. That's why in US now, it has become the regular mammogram has become redundant. Now we are doing a more advanced form, which is an advancement of biotechnology by itself, which is 3D mammogram or tomosynthesis, or we will do a simple ultrasound in addition to the mammogram. So I'm just requesting everyone that if you know of anyone, who has a lump going through the mammogram, please advocate, please tell them that if you have to ask your doctor if you have dense breast or not. If you have dense breast, then definitely go for an additional screening, which is very simple, which is ultrasound, and that will really help us to detect the tumor. Next slide, please. So now, when I thought of first, starting Breast Cancer Hub, resigning from my job, career, and everything, my first thought was I'll be going to the villages in India, and even it is a culture-induced inertia. Even when I asked my mom that would you like to go for a mammogram, she wouldn't go. She said, I don't want to go to a cancer hospital, though I belong to a family of physicians. So then I thought, what can I do? In US, what about the younger women? Now in India, US, worldwide, younger women. I met women in India, 22, 25, 29 with breast cancer. But how did they detect themselves? Through breast self-exam. Here also the younger women lives are saved because doctors do clinical breast exam. So what doctors are doing, I replicated the same thing, collaborated with the oncologist in US, and we built this breast self-exam card. One side is for women and one side is for men. Now, when I thought of India, like I'll be going to the villages, if I'm giving the card in English, this will never work. So now I have it because of our volunteers, but everything we do is free of cost, and all our volunteers in our organization. So we have it now in 18 languages. So next slide, please. Now, if you may ask me, like in the villages, even the women and men, they will not know to read and write. But you see the small kids over here holding this paper. I give the responsibility to the small kids. You read it out to your parent. But that doesn't mean that, what happens next. Even in US, they love the breast self exam card. So what happens next? I take accountability. Everywhere I go, everyone has my contact number. They reach out to me. Even if I'm going to the villages, the village head will be there. They will know my WhatsApp number. They will reach out to me, and then I connect them to the local healthcare centers. I follow up. We have adopted like 8 villages total, 2 villages already done, but we go now door-to-door screening, and we do the oral screening also because of the tobacco issues, breast screening also, and we collaborate with the healthcare center so that we take accountability of everyone wherever we are stepping in. And we saved around 5,000 lives just with a small paper. I know it's not a high-end paper, it's just breast self-exam, it's not a bombastic word, but trust me, this small thing will save our life when we know the correct way to do it. And I'm not going into explanation because I don't have time, like how to do the actual protocol, but if ever you need any help, I'm always there. Next slide, please. Now, a very important topic is family history in genetics, because I know you all are very profound with genetics, so I don't have to go in depth at all. But when in a family we have ovarian cancer, breast cancer running in the family, or any type of cancer, we have to be aware. We have to go for a genetic testing, which is called BRCA1, BRCA2 testing, and also they do a panel. So panel in the sense there'll be low, medium penetrance gene, low penetrance genes, which may be prevalent. Now, many cases, what I found is that we always forget the man of the family. So the men need to do the BRCA testing and the other genetic testing equally if the person's mom, aunt, they are suffering or they have breast cancer, victims. Because for the men, it's not only they're prone to breast cancer, but they're also prone to other type of cancers, for example, prostate cancer, digestive tract cancer, and other cancers. So please be aware and spread this word about the genetic testing. And I know like a lot of times insurance doesn't cover. It's pretty expensive. In that case, when I'm going to the villages, I don't talk about genetic testing. But what I say is, if it runs in the family, be vigilant. Just be more careful about it, so that you're on top of it, and then any issues, you immediately reach out to us. So that's how we can handle the situation. So it's not about poverty, it's not about education, it's about awareness, that simple steps will save our lives. Next slide, please. Now, a very important, so we all know that 30 to 50% cancers can be prevented, but I'm not going to talk about that. But now I'll come into more detail, little bit of molecular level, is that inflammation. We all are very profound with this word inflammation because in COVID also we are talking about inflammation, the inflammatory influx in the lungs and what causes the final collapse of the lungs. But with cancer, inflammation plays a vital role. So my work, this particular work, we discovered the molecular mechanism between arthritis and breast cancer. So how, like, because of arthritis, the travel, the metastasis with breast cancer is significantly higher. And my work got press release in 2012 in American Association of Cancer Research for this discovery. So I would like to bring into limelight a little bit on this factor, which is very critical. Now, inflammation plays a dual role. Without inflammation, we cannot survive. But when inflammation goes out of control, that's not good because inflammation helps in the progression of cancer. So what happens is when the tumor is there, it actually, the tumor microenvironment also produces a lot of inflammation going on around the tumor microenvironment. Then also, there are other pro-inflammatory cytokines like IL-17, IL-6, and also there are other factors like macrophage, colony-stimulating factor, VEGF, and other elements like cytokines which are highly upregulated. So what happens is when we have arthritis, which is inflammation of the bones. So next slide, please. So when we have inflammation of the bones, what we discovered is that these pro-inflammatory cytokines are severely high, and that helps in the drawing of the the cancer cells towards the bones, which is anyways the favorite place for the breast cancer cells to travel. That becomes much faster. And the experiment, what we did was we had this transgenic model of mice, which we called SKG mice. They developed spontaneous arthritis. In those mice, in tons and tons of experiments we have done, in the mammary fat pad of the mice, we induced breast cancer cells, like highly metastatic and aggressive type of cells, so that we replicate the microenvironment. So when we did that, next slide please, what we observed was that so with arthritic model versus non-arthritic model, next slide Tanya please, with arthritic model and the non-arthritic model, the inflammation in the bones also increased a lot with breast cancer. That means the breast cancer cells by itself, it is traveling faster, plus it is creating a microenvironment even at the bones also, so that there is more and more inflammation in there. And we also found that you can see the lesions over here in the lungs. We also found that the lesions, the metastasis, significantly increased even in the lungs, which was not telling with our hypothesis because we were always thinking about the bones, but we always forget that there is an underlying inflammation and that actually affects the lungs also, and that's why the metastasis is significantly higher. Next slide, please. So we ran the same experiment, in another. So in the bones also, we found a similar trend. The metastasis was significantly higher with the arthritic model versus non-arthritic model. Next slide, please. And then what we found was that what is actually causing it? Next slide, please. Tanya, next slide, please. Yeah, thank you. So we also replicated this in another model of mice which developed spontaneous breast cancer in which we induce arthritis. So basically in different models, like arthritic model, we induce breast cancer. Another model was spontaneous breast cancer model. We induce arthritis. In both the cases, we found a similar trend. That means that with arthritis, the breast cancer metastasis significantly augmented. Next slide, please. Now, we tried to go deeper. So we ran many experiments, we targeted different cytokines, like IL-17, and we did a lot of treatment therapy, and we found that when we reduce the inflammation, automatically the metastasis also significantly reduced. Now, one pathway that was very significant, next slide please, which we found was that, that is very important, and we never even think in that direction, was that the mast cells. The mast cells is very common. We all are so familiar with mast cells. The mast cells have a receptor which is called the c-KIT receptor, and the breast cancer cells, Next slide, please. Breast cancer cells have a factor which is called stem cell factor. Now, what we found was that it is like a lock and key mechanism. Next slide, Tanya. So it is like a lock and key mechanism. And what we discovered, if you see here the red stains on the cancer cells on the left side, these are the breast cancer cells. And then the stem cell factor is all red. That means that all the cells were expressing the stem cell factor. And then in the bones where there's huge inflammation, we have the mast cells. And then the mast cells have the receptor, c-KIT receptor. So when these breast cancer cells are traveling, automatically it significantly pulls it towards the distant organs, which are the bones, the lungs. And also it's the other way around, because even in the tumor microenvironment by itself, the stem cell factors are attracting more and more mast cells. So that way the inflammation at the base also is significantly higher. Next slide, please. So when we discovered this mechanism, what we try, and you can see the stainings here. These are the stainings of mast cells in the lung. The top one is the lung, and the lower one is the bones. That means, and on the left-hand side, you see the non-arthritic model of the lung and the non-arthritic model of bone. And you can see it explicitly how on the right-hand side, the staining is much more because there are more number of mast cells with the arthritic model. Next slide, please. Now, how do we target that? What we did was we did a treatment with the anti-c-KIT receptor antibody. So when we did that, we found that there was a significant decrease in the metastasis when we actually target the mast cells. So that way, this was a great finding because now we, next slide, please, Tanya. So now we know that, if we are actually targeting the mast cells, the automatically, like the inflammation is decreasing, and that is reducing the metastasis from the breast cancer cells towards the bones or towards the lungs. Next slide, please. So based on this, the conclusion that we drew was definitely that inflammation is playing a significant role. And now when it comes to inflammation, it's not only about arthritis, other different chronic diseases that we are having. So when we have cancer, any type of cancer, we always target the cancer. We always forget that, other diseases become secondary for us, be it diabetes. Even if we have diabetes, it is underlying inflammation. Even if we have obesity, the weight needs to be controlled. That is also underlying inflammation. So we really need to curtail the inflammation in the body, which we can do in many different ways, through diet, to exercise, to relieving the stress, and that is another chapter which you all are aware of. But inflammation definitely plays a vital role basically with cancer. It's a very significantly, we are trying now, because breast cancer is not of one type. So many types are there. Receptor status plays a role. The aggressiveness of the tumors play a role. So it's very important that we really have a control on the other types of diseases too, because that's what now with the oncologist I'm working on is on the clinical scenario. So because it's very difficult when I'm trying to, even in US, when I'm trying to work with the oncologist, they are not asking the patients what other diseases you have, because that column is not there. The data doesn't exist. So now we are trying to penetrate there, requesting, please take a look. Even when I'm in India with different oncologists, and they then tell me, yeah, we really see a profound difference. We see a drastic, significant difference when we are trying to target the underlying diseases, because that actually helps in controlling the progression of the cancer. Next slide, please. Tanya, next slide, please. Yeah, this is my second last slide. So basically, in this, in this particular talk, what I wanted to go from was from the screening, why it's important, why the basic fundamental change that we need in India, and a platform like this is really amazing, which can be taken forward by you all, by everyone, especially the students, is helping to break the breast taboo. Because because we have lost many moms. I'm going to the villages and they tell me okay, every time I go, it's 100% death rate. We cannot lose any more moms. Like the saddest story was last year I went to a very remote village in Assam. One girl after my session, she was crying. She told me like, why didn't you come to my village last year? My mom just died of breast cancer because she was shy and she did not know. So I couldn't save her, but I told her no other moms will die from your village. And that's how we adopted her village and we are going door to door. So it's the simple thing that makes a difference. High-end technology definitely is needed for the diagnostic, but we have to detect early because early detection is the key to any type of cancer. And early detection, 100% cancer is curable. Other diseases are chronic, diabetes, kidney disease, but cancer is curable with a positive attitude, because we should not fear cancer, and with a good support system, palliative care, cancer is 100% curable. But also we have to keep a control of the other underlying factors. Last slide, please. Next slide, please, Tanya. Saying that, I would like to really thank you for giving me the opportunity. Thank you to National Biotech Summit, for inviting me here, and so that I could present my views of so many important things that I feel that if we all come together, we can really bring this change, because together is always, working together is magical. Thank you so much.
Thank you, ma'am, so much for really great talk.
Thank you.
Yeah, so we have one question by Praveen sir here. I'll just ask that. He's asking, are you doing any clinical work in this area?
Yes. So if you are talking about like, so I'm doing a lot of epidemiological research study in the untapped area. So villages we have adopted, but I have also, I'm working with many oncologists on the looking into the inflammation part. So with the research proposals that we have good research proposals that whatever we have found out, so if they can, so definitely we are working on the clinical aspect too. Yeah, absolutely.
Okay, but have you done some cases, 10 to 15 cases to see the what is the outcome is coming in this?
So we have some data that, but that with n number of 10, we cannot draw any conclusion. But the data definitely represents that with inflammation, and it is already proved. The National Cancer Institute has already proven it. There are many published articles now, so we are showing that with inflammation, the progression of cancer is significantly there. But with breast cancer, with our study, we have data where we are seeing that inflammation is there, and the cancer progression is definitely on the like on the increasing side. But one good thing that happens is, those patients who are already arthritic, they are already in the anti-inflammatory medication. Like they're already having a lot of NSAID or maybe IL different interleukin injections. That kind of works in the other way also. That way it kind of helps with killing the inflammation, which when we are having cancer, that kind of, help, it basically helps each other. But if we are leaving it untreated, then that's the biggest problem that we are facing.
Okay, thank you.
Thank you, sir.
It's a
Okay.
good work, and
Thank you.
need to have a collaboration. We'll see.
Absolutely. I'm always open for
Okay. This
collaboration.
is a foundation, private foundation, or
No, it's a totally non-profit organization. So I'm the founder and
good?
president of it. So I resigned from, I was a research professor at University of North Carolina. So I resigned in 2017 so that I can work solely for this.
Okay,
Yeah,
good.
it's a
I am
totally
also
non-profit.
okay. Good, very good.
Thank you. Thank you.
Yeah, so we have one more question from Mohini ma'am. Asking, I just wanted to ask whether if there is any other kind of autoimmune inflammation there in the body, then also it is directly related to cancer. And if this relation is only with breast cancer or with any other type of cancer also.
Yeah, very good question. So IBD, inflammatory bowel disease, so that also like has shown a relationship. And any type of cancer with inflammation will play a role. So after our discovery, then like in Mayo Clinic, the other lab, when they saw us working on this, they started to work on asthma. Because that's again inflammation, and they also were finding the similar kind of results. So there are a lot of results which shows the inflammation, but there are a few contracting results, and that is happening because of when we have all these inflammatory conditions, we are already on very advanced stage, like like lot of good resources, lot of patients who are very proactive, they're already in anti-inflammatory medications, right? So which kind of helps them to negate the fact that inflammation will have. Like yesterday only someone from Thailand reached out to me, they have severe allergies. So what can we do? Because that's also inflammation. So I'm going to talk to
Ma'am,
you.
actually, Mohini ma'am is asking about autoimmune inflammation, not
Yes,
IBD.
yeah, in any autoimmune diseases, autoimmune inflammation will play a role, definitely. So, but each type, you can look it up because my work was on arthritis, right? So I cannot say confidently about others, but in general, any autoimmune disease which is related to inflammation that will affect with the cancer progression.
Okay, thank you, ma'am, so much for the great talk. Now we'll move ahead. So our second speaker would be Dr. Amit Kumar, professor from Amity University, Haryana. We welcome you, sir. You can now
Okay.
start. Yeah.
Good morning, everyone. Uh, and I would like to thank Jack's Foundation for giving me an opportunity to give a talk here in National Summit. So I will start presenting now.
Yes, ma'am. Yes, sir.
I hope you can see the slide.
Yeah, it's loading. Yeah, we are able to see the slide. Yes, you can continue.
Okay, so I was told that majority of you will be the participant will be the students. So I have tried to make it as basic as I can, since it is a broad topic which I have selected for the talk. It's non-coding RNAs as biomarker in cancer, because my lab is more interested in non-coding RNAs, especially microRNAs and link RNAs. Uh, so just to give a brief introduction about biomarkers for the student, biomarkers are molecules which indicate normal or abnormal processes taking place in your body, and it may be a sign of some kind of disease or any condition. And molecules such as DNA, proteins, or hormones, they can act as a biomarker, and then you can get some information from them regarding your health. Now, biomarkers can be produced by the cancer tissues also, or by other cells in the body in response to the cancer, and they can be found in mostly bodily fluid such as blood or urine or tumor or solid tumors also. Now, what kind of questions can be answered by the cancer biomarker? So it can be either prognostic, like it is likely to develop this cancer. In diagnostic, so you can know what kind of cancer it is. Predictive, is this the optimal drug for my cancer? And in pharmacodynamics, you can think about like what is the optimal concentration or the dose of this particular drug for my body. And recurrence is when will the cancer return again. So these are few of the steps to evaluate and validate new biomarkers. Uh, Step one is to identify the promising biomarkers, evaluating the ability of the putative biomarker to detect disease by clinical assay. Then you have to determine the ability of this biomarker to put into the preclinical disease. Then you have to perform the screening to know the extent and characteristics of the disease detected by putative biomarker. And then you also have to check whether how much of them is a false positive, and you have to remove it. Now the other last one is the random trials have to be conducted to determine the putative biomarker in decreasing the mortality rate. So these are the few general steps which have been followed. Now these are few of the examples which are already there in the literature, and people have been using these set of the genes as a biomarker in cancer. So if you see on the right hand side, HER2, BRCA1, as earlier speaker have also told. So these are the biomarker for the breast cancer, for KRAS, P53 mutations in the non-small cell lung cancer. So similarly, KRAS is also acting as a biomarker in colorectal cancer, and BRCA2 and CTC are the genes which are biomarkers for the prostate cancer. Similarly, there are some of the targeted therapy against these sets of cancers also. but my main interest is the non-coding RNA. So if you see, the majority of the cellular RNA does not code for the protein. Out of the whole genome, 80% is the transcribed genes. Okay, out of those transcribed 80%, 97% is non-coding. So non-coding are those which does not code for the protein. So you does not get the protein from these sequences, and only 3% of the transcriptome is coding. So it codes for the protein, and proteins are made. Now, if you see, that means human RNA is approximately 97% non-coding. So what is this? They were treated as a junk. So what are the these non-coding do in the body if they are present in such a large number? So these that's why lot of interest has now been come into the RNA field, and people have started studying these RNAs also. So if you see, these can be classified into two broad topics. Either it can be small microRNA or long non-coding RNA, link RNA. So microRNAs, to make life easier, people have just classified them into the size. So the RNAs which are less than 200 nucleotide are classified as a microRNA, and they are endogenously produced by endonucleases, and these sequences are very well conserved also. Approximately 2000 microRNAs have been already identified. And in case of the link RNAs or the long non-coding RNAs, those who are more than 200 nucleotide, and it can go up to 10 kb also. And mostly these are 5' cap and polyadenylated in part, and also splicing is done. And these sequences are poorly conserved. And till date, if you see, more than 30,000 link RNAs have been identified, which are more than the protein coding proteins also, or the genes which are coding for the proteins. So the human genome has identified approximately 23 to 25,000 of the genes. And if you see, there are already 30,000 link RNAs have been identified. Now, this is just a timeline of the discoveries of the RNAs. So in 1869, if you see, nucleic acid were discovered, and this first long non-coding RNA H19 was discovered in 1989. So it was so long back. And another link RNA GIST was also discovered in 1990. And the microRNAs were discovered rather late in 1993. So if you see, these link RNAs have already been in the research field from 1989. Now, coming to the microRNA biogenesis, how these microRNAs are formed, just a brief introduction of the formation and functions of the microRNA. So microRNA are give rise to the primary microRNA from the microRNA gene, or they can be from the intron also. So they can be intronic also, and from the any gene also. So these microRNA, primary microRNA, are then cleaved by the DROSA and DGCR8, and then they form the precursor microRNA, which is approximately 75 to 90 base pair. And the primary microRNA is usually 150 base pair. And then this precursor microRNA is transported outside from the nucleus to the cytoplasm through the exportin 5 and RAN GTP. And these then are cleaved by the dicer enzyme, which cleaves it into the microRNA duplex, and one of the base pair which has less stability of the 3' end is degraded. And then this microRNA is formed into, comes under the risk complex, and there it binds to the target microRNA. And most important of this microRNA are the seed region from the 2 nucleotide to 8 nucleotide. So those are the most important aspects of the this particular microRNA, and then they can bind to the target RNA, leading to the cleavage or translation repression or deethylation. Now I am giving just a brief summary of how these microRNA are used as a biomarker, because a lot of research has already been done, and people have shown a lot of microRNA as a biomarker in different cancers. So I am giving you a general idea that from the microRNA biogenesis to the release of the microRNA, these microRNA can be detected in microvesicles. They can also form a protein complexes. Then there are lipoproteins or apoptotic bodies are there, and then passive release into the body fluid. So you can take out the blood, and there you can detect the microRNAs in particular cancer. Now, you can extract the small RNA from serum, plasma, urine, or milk, or other body fluids. And there are already a lot of tools to quantify these microRNAs, such as qPCR, microarrays, and nowadays high throughput sequencing is being done. And these, after detection, either you can predict as a diagnostic marker or the prognostic marker. And then you can take the clinical decision whether you want to do a personalized therapy or the target identification and development. Just for example, microRNA 122, which is a liver specific microRNA, is already in clinical phase trial 3. Now, these are few of the examples. If you see, already 464 clinical trials we are started investigating the microRNAs, and only 163 we are associated with the cancer, and rest we are from non-cancer. And out of those 163, 52 trials are already in the phase 1 to 4. And of those 52 trials, if you see, only 3 trials validated a specific particular microRNA as a circulating cancer biomarker. 22 trials have already identified new circulating microRNAs, and then 2 trials using microRNA mimics as a treatment for the primary tumor. So this gives you a brief idea that microRNAs are being successful in being predicted as a biomarker. Just a brief introduction for the link RNA biogenesis. So these link RNAs, as I told you, are synthesized from the same link gene. So it can be intronic also, it can be intergenic also, or it can be from the introns also. So this is a brief idea of how these are being synthesized. So they can be also a natural antisense transcript. As a result, they can also go and bind to the genes and target them. Now, these link RNAs can be classified into 3 important aspects. So they are scaffold, decoy, or guide. So and then this is the functions of the link RNAs. So they can induce chromatin remodeling, transcriptional interference, or small RNA precursor. And they can also modulate the protein activity, or by binding to the specific proteins, or alter the localization of the protein. And they can also become a part of the complex of the protein. They can also act as a sponge for the microRNA. The sponge, the meaning of the sponge here is they can bind, since these link RNA are very big in size, so they can bind n number of microRNAs inside it. So they act as a sponge. And then they can also modulate the splicing pattern in the cells, and they also can generate endogenous siRNAs, which results the virus response also inside the body, because the body gets confused for the external virus. Now, what are the characteristics of the link RNA which make it as a biomarker? So some link RNAs have high specificity and sensitivity to the tumor process, and these link RNAs can be sampled in body fluids with relative stability. And also, some link RNAs are useful as a diagnostic and prognostic tools in oncology, since they behave as a predictor for the survival time and risk of metastasis. So this is very busy slide. I will just give you a brief idea that these link RNAs act as a major hallmarks and enabling cancer characteristics. So they can evade, help in growth suppressor. They can enable replicative immortality. They can help in promoting tumor or inflammation, genomic instability, and evading immune destruction. So these link RNAs can also act as a RNA primer. They can promote metastasis or reduce the metastasis also, and they can also resist cell death. So if you see, these link RNAs have been involved in lot of cellular processes. And then people have also designed effective management for diagnosis. So what they do is, this is a general protocol. You can take the body fluid either from the saliva, serum, or the urine, or the stool, and then these are the targets. Like you can either select DNA, microRNAs, no RNAs, or snRNAs, or the link RNAs, and then you can perform the screening. Then you can classify them into the low risk and high risk, and then you can give them the personalized therapeutics and monitor the disease. So some of the clinical implications already there in the labs. So in the prostate cancer patient, they collect the urine sample and extract the RNA from the cells which are coming in the urine, and then they assay for the PCA3 expression, and then they correlate this expression with the link RNAs also. And then they other is what they do is they have this RNAi based therapy or the small molecule based therapy, where they take the link RNA and label them, and then inject through the intravenous administration, and then they see what is the effect of this particular link RNA in the cancer. The other is they profile the germline prostate cancers and look for the SNPs, single nucleotide polymorphisms. So these are the few aspects where these link RNAs have been involved in the biomarker, and lot of research is still going. So my lab interest is in unraveling the molecular mechanism of link RNAs involvement in glioblastoma, triple negative breast cancer, and ovarian cancer, and metastasis in ovarian cancer. So I currently have 4 research projects, and from different granting agencies, and my lab is focused on evaluating the mechanism of these link RNAs in these 3 cancers. So this is a small glimpse of the department or lab. We do have very good research facility here, and there is a link. If you we have the central facility, and we outsource these facilities also. And thank you for your attention. Hello.
Thank you so much, sir, for the really great talk. Now we have actually one question by Anmol for Lopa ma'am, if she's there. Lopa ma'am, are you there? Okay, so I guess ma'am is not there now. So maybe ma'am will answer the question later, Anmol. So we'll now go ahead with the next speaker. So our next speaker for the day would be Dr. Pulok Mukherjee, Director, Institute of Bioresources and Sustainable Development, Manipur. So I welcome you, ma'am. Ma'am, are you there? One second. Okay, so till then we have got a question for Amit sir by Jyoti ma'am. Any clinical with respect to non-coding RNA in case of cancer therapeutics?
Yeah, so I gave the example like microRNA 122 is already in the market now, and it is a liver-specific microRNA. So it treats the liver cancers, and a lot of other, as I told in the clinical trials, that already there are 22 to 23 microRNAs are in the phase 1 to phase 4. So soon they will also come in the market. For the long non-coding RNA, they are still new, and a lot of new research is still under process.
Okay, thank you so much, sir. Now we'll have Pulok sir here, I guess. Yes, sir.
Can you allow me to share my screen?
Yes, sir, you can share. Yeah. There is a present now option down there. Just click that,
Okay.
you'll be able.
Yeah, present now.
Present entire screen.
Your entire screen. Okay.
Then just open the PPT. I guess it should be done.
Can you see the? Can you see my slide?
No, we are not able to see that. It's not started presenting it.
Can you see it now?
No, sir. So you have to click on the present now there down.
Yeah, I'm clicking there only. I think you have to permit me something like that.
No, sir. Like last time also, Amit sir directly did. So I guess you can do directly. There's no permission required. You can
So
directly do.
present now, then entire screen. Okay, your
Yeah,
entire screen,
yeah,
right?
entire screen, right.
Yeah, I'm doing that.
Tanya, are you there?
Sir,
Can you look into this?
sir, you have to select the slide first.
Okay, I have done that.
Tanya, are you there? Hello.
I've opened the slide and then
Yes.
share, right?
Yeah, so if you have Tanya sir ka presentation, you can maybe present like Lopa ma'am did, maybe. I think that should also be okay, sir, I guess, if you are fine.
Aditya, I don't have a slide of sir actually.
Okay, so can you just guide, sir?
Yeah, yeah. So in the
Can you see the
bar
slide
button,
now? Can you see
yes,
the
sir, we
slide
can.
now?
Yes, it
Yes,
is
yes, now
visible.
it is
Yeah, so it
loading.
was
Yeah,
stopped.
yeah, yeah. Now it is visible, sir. Just make it full screen. Down there is an option. Yeah. Yes, now it's
Can,
visible. You can continue. Yes.
can you, hello?
Yes, sir, it is visible. You can continue.
Okay, at the outset, I would like to thank the organizers for inviting me. Greetings from Imphal. As in, you know that we are working on the natural resources of Northeast. In fact, I came to this institute as director in February middle, and from March onwards, you know that we are seeing the world in a very different way, and in this pandemic situation. So I'll tell you some of those context on where what are the field on which you are, which way the things are moving in the area of drugs from nature, where evidences for validation is one of the major thing. Uh, I was working as a professor at Jadavpur University. My main field of work is on natural products drug development, specifically traditional medicine inspired approaches. Now, say one thing, this COVID-19 scenario has really taught us that we know the world in a very different way, and we know our drugs from natural resource also in a very different way. You see, starting from politician, bureaucrats, scientists, everybody talk about the medicinal plants and its therapeutic value as immunity booster and so many things in this particular area, and science is progressing in a very different way. So I'll tell you some of those contexts here on which there are enough scope to work, and there are a lot many things to be done yet for the evidence-based validations of our own system of medicines, what we are practicing from years together. So a brief about the Institute of Bioresource and Sustainable Development. It is from the it is under the Department of Biotechnology, Ministry of Science and Technology, Government of India. We have several other centers in Sikkim, and then one node in Meghalaya, another node in Mizoram in Aizawl, and on which we have been working on the main motto of exploring the bioresources and their conservations, preservations for sustainable use, for the development of the bioeconomy of the countries, particularly from the northeastern region of our country. Coming to the topic, when we talk about traditional medicines and validations, you see that we know that there are two type of systems persist in our traditional medicine concept. One is the codified documented system, like we have Ayurveda, Chinese has DCM, Japanese has Kampo, and so on. And another one is the non-documented system of medicines, where it is not documented like Ayurvedic Sanghita or pharmacopoeia or formularies, but those are being transferred from generation to generations in a very, very different way, which is the Lok Shastra Parampara. Both of these contexts has a very definite perspectives, and these perspectives depends on the concept how we take our traditional ancestors medicines. But I believe this is all, whatever the research work, anything and everything what we are doing, these are all traditional medicine inspired approach, and this traditional medicine inspired approach is mainly based on our ancestors concept. So we learn from our ancestors, learn from our nature, and then we do science where tradition meets the innovation. And if we work on that particular context, then two things. One is ethnopharmacology, ethnomedicine. That definitely has a very different context. And another context is the exchange of knowledge, or rather developing platform for knowledge exchange, empowerment, and so many other things with the traditional healers, who also is contributing to a high extent, and where through our institutes and through my previous work we are trying to develop a benefit sharing with the traditional healers, bringing their drugs into the main platform, which can be lead for so many things which is being practiced now. Now coming to the concept of ethnopharmacology, on which our main context of work depends, is that this ethnopharmacology is one of the traditional health practice which is being practiced from years together. The word was coined in 1967 with the study of hallucinogenic plant mainly, but even before that, if you consider about Ayurveda, we have been practicing it from years together, maybe more than 1,000 years, and it particularly concentrate on the study of the medicinal plants or their therapeutic benefits. And one of the context here is it is grouped under large branch of science and technology discipline. Say like it involves anthropologist, pharmacologist, biochemist, chemistry, then many other discipline. And in fact, nowadays, what on the working pattern, what we believe, that we need very linguistic person to develop those traditional system of medicines, because in the multilingual country like India, we have a lot of resources which has not been translated into English or some other language, and are losing so many things. So friends, when we talk about this particular context, so this is a very big thing where the integration is required, and integrative medicines, ethnopharmacology is where history tell the future. Yes, if you see the drug discovery history, you see about aspirin development, a simple molecule which was developed in 1890 for analgesic. It has been used for so many other uses afterwards. The single molecules from Salix alba wood, say like it is being used in pregnancy complications, in stroke, even in cancer. And like that, if you see that the development of drugs for cancer, again, say we have been working with so many things on the synthetic molecules, but it is the nature which has given some answer for cancer, including taxols from Taxus bravifolia and so many other plants. And even if you think about the very modern development on the anti-malarial drug, again the same story that we had quinine from Cinchona, and now we have artemisinin from Artemisia aqua. So when we talk about that we require the integrated approach for development of medicinal plants from traditional medicines. This integration, it requires from selection of the plant to its high throughput screening, profiling, then its standardization, quality evaluations, kinetics development, regulatory study, pharmacovigilance, and so many other things. One thing very interesting is here that we talk about in a drug development, the clinical study. But here, the clinical study has been done for years together by our ancestors. And that is the beauty for which that the working in this particular area is somewhat that we are rediscovering something which was set up by our ancestors long back. That the integration can lead us for development of those drugs to a more scientific way, or rather documenting those drugs in a more scientific way, which is very much required. And I'll give you some of those examples in this next few slides on this development of those the rediscovery process, where profiling, the chemical profile marker analysis is a very important aspect. So what do we believe in science, that there is the molecules in whether it is in a synthetic or in a natural product or whatever, the chemicals it works. And those chemicals give some biological activity by interactions with the enzymes or in our body, and by that way we get the effect, therapeutic benefit. And definitely there are some side effect as well, and then we are effect or side effect that nullify with some other component. This particular context, if we come to the drug discovery perspectives, you see there are two very prominent contexts. One is the reductionist approach, where we are getting one single molecule like Taxol, artemisinin, aspirin, and another one is the synergistic concept, where we go on that evaluating the synergy of the molecules together present in that particular plant. But I believe that this establishing synergy is one of the major thing what whole our medicinal plants or the traditional medicines need to be. And I'll tell you some of those contexts on which we have been working. Specifically, when we want to talk about the synergistic development, we need to know the chemical profile of the plant, and that is possible by marker analysis. And this marker analysis is a very simple task we can do by chromatographic study with HPTLC, high performance thin layer chromatography. What has been done here in endographic speniculator, taking endographoloid as the marker, and we make the things in that way for establishing the concentration of endographoloid present in endographic speniculator extracts. This endographic speniculator is now very potent, even have been proved to be used for several contexts in this pandemic situations for immunity boostings or even as antiviral as well. And then when we go on, so if you see, there are several other examples on that, where we have been that like Glycyrrhiza glabra with glycyrrhizine is the marker, and then with Piper longum, and so many others on that, on which we have been working. Several other scientists in the country and the world, they have been working on that. And there are development, so many, like say we have Indian pharmacopoeia, where this profiling has been included. Even other regulatory bodies, they have accepted those things. And in India, we have even the phytopharmaceutical law, where we can establish those particular extract as a drug while we are establishing their chemoprofiling, and then that can be prescribed even by the modern medicine doctor as well as per the drugs rule. Beside that, we have the Ayush route, where we can develop the drug based on the Ayurvedic pharmacopoeia. And if you go through the United States pharmacopoeia, they are this based on this their dietary supplement and other rule. Several things has been made based on this profiling and chemical systematics. What I'll tell you in the metabolomics in later few slides. Now the question is that, say, like, it is not that identifying the marker is the main target. It is that we have to validate our own natural resource, our own medicinal plant on this particular context of their chemical profile, to know about the purity of the purity of the plant, or whether the variations in the constituents need to be studied. One interesting example, say, like, you may say that, okay, for single plant we are getting a marker, but when you talk about traditional medicines, we are using polyherbs, so many herbs together we are mixing. Then what is the answer there? Because like if a single plant, like say if you see kalmek here itself, you see there are more than 8 constituents we can see in one plate. Maybe if we change the solvents, there may be the changes. Now if we mix that like that 6-7 plants, that means what we are having around over 30 or 40 ingredients together. Then how to do that? Yes, there is the answer. Like say, if you think about a formulation called Triphala, which was mixed thousands years ago by the rishis of the great, I should say, the greatest scientist of our country, who were not knowing our present hyphenated techniques like LCMS, MS, GCMS, HPLC or other things, but they mix these 3 fruits, knowing that there are some very potent therapeutic benefit. And this Triphala, as you know, that 3 fruits mixed together, and this has its own constituent nature, and then it has its own therapeutic benefit as well. And this is one of the highest selling product in our country, from within the country. Even export potential is too high. And when we work on that particular context, what we find that, interestingly, these all 3 fruits contain the same biologically active marker. that is the biological reference standard, what we call in our pharmacopoeial terminology, that BRS, not even one, there are two, ellagic acid and gallic acid. As you know, both of them are very potent antioxidant and very potent rejuvenator, and that is why Triphala is the one of the greatest rasayana from Ayurveda what our country have. And in fact, those constituents present in all these three fruits in a very potential ratio, and which presence have been established with chromatographic studies. Then there are a lot of work has been done. In fact, the cytochrome P450 enzyme inhibition study has proven that Triphala is safe to use. Now you see the science behind traditional medicine, specifically Ayurveda. This is the greatest science. As I told, that we are not discovering something, just rediscovering or re-establishing the fact behind establishing the science on the particular context on which we have started, we have been working in this particular area. And one context we must, another context we have to think about the interaction study. Like say, there are a lot many things happens nowadays on the herb drug interaction, herb interaction. And what I feel that this herb drug interaction is very pertinent in our country specifically. Like say, when a person is suffering from diabetes, he is taking the sulfonylurea as prescribed by the doctor, and together with he's taking that Karela juice, Lokika juice, and so many other things. And for that reason, sometimes there are a lot of evidences of hypoglycemic shock. So we need to know about those herb interaction, herb drug interaction, and document those for the benefit of the society at large. So friends, the need of the hour is the systematic approach for validation of our traditional medicines. This validation starting from societal context to the therapeutic context, chemical context, biochemical context, everything's together, which can uniform the use and the chemical quality control standardizations of those particular contexts. This was the effort what was made in 2015. Now this year also we are just revising this document. This document was published by Elsevier Science with 21 countries people wrote on this particular context, and I believe that that you can access that one and you can get a lot many ideas on these approaches for validation of traditional medicine on which way it should be. Coming to the context, that's when we go on with the chemical context isolation evaluation, there is another important issue is on the how best we can evaluate the therapeutic benefits. There are a lot of enzyme inhibition assays, in vitro assays, in vivo assays, what you have been hearing for yesterday onwards, even today, a lot many speakers have highlighted on those particular contexts. What I believe that if we go on a very enzyme inhibition study or in vitro or ex vivo studies, we can come up with several of those particular contexts. Here is example of acetylcholinesterase inhibitors, which is for Alzheimer's disease. And in the Alzheimer's, so like when we talk about this specific context of developing a drug, then this there are a lot of molecules which are here, which have been which are being used for years together for different disease, specifically with the effect on the central nervous system. We screen many of those plants with several specific objectives on these acetylcholinesterase inhibitors. As you know, this acetylcholinesterase is the enzyme which is responsible for breakdown of acetylcholine, which is a neurotransmitter in the learning and memory process. Now, when we talk on those particular contexts, that acetylcholinesterase inhibitors, they can lead us for development of drugs for Alzheimer's. And here, when we work on those many of those plants, then they have potent acetylcholinesterase inhibitors and which have a scope for development of drugs for Alzheimer's disease. On that, we have been working for several years with the involvement of scientists from UK and other countries as well. And we have got several of those contexts which need to be evaluated specifically for development of drugs for Alzheimer's. When we think on those contexts, like I show you one disease like that, lot many specific context of disease is there while you study the enzyme inhibition. Here, one thing the context on which on validation, what I am talking today, that main and major context is the metabolomics. And these metabolomics tells you about the constituents, their interaction, and in fact, By that we can establish the mechanism of study. And these metabolomics can be targeted or untargeted secondary metabolite identifications. It helps in quality control of medicinal plant, chemotaxonomic and phylogenetic characterizations, identification and quantification of adulterants, metabolite target analysis, metabolite profiling, pharmacological and pharmacokinetic study. Now, I'll give you some of those examples here. Say like, this is Bacopa moneri. This is one of the very well used medicinal plant in the concerning with the central nervous systems, in the learning in memory. It helps, and where we can apply this metabolomics, where it is like, say, Bacopa moneri, bacoside is the constituents which is biologically active, but this bacoside tablet, it never works as like as Bacopa extract. So there are the constituents which synergize the activity, and we need to know them, what by which we can establish their efficacy together with the synergism. And friends, when we talk about that, the interesting thing is that in the tradition, when you see, there is a terminology called samyoga, and samyoga, the knowledge of synergistically combining ingredients for maximum safety and efficacy. Now, when the whole world is talking of synergy, our traditional system has already prescribed samyoga as the synergism, and this synergism play a major role in therapeutic efficacy of herb and herbal formulation. What I believe that this samyoga or concept of synergy, what is there that really relates with our own effect on the use of food, medicine, or medicinal plant as food, as our food, or as our those plants as our medicine as well. This is the concept what need to be, and how we can. So this is the metabolomics linked with synergy and network pharmacology can help us. Take the example, say like Karela. It is a very well used medicinal plant, as you all know. Now, when we are studying this pancreatic lipase inhibition kinetics with LC-Qt of MS-based identifications of this bioactive constituent from Momordica, then you can see that those constituents together, the study explored that the phenolic compounds present there serve as the functional food component which address the obesity-related disorders. And coming to the example, another example on Trigonella, Phonium grassam, as you know, methi, it is a very well used herb throughout the country. And when we work on that, you know that this is used for hyperlipidemia and hyperglycemia. And when we do go on the LCMSM study, identified over 13 compounds, and those 13 compounds, when we relate with the network pharmacology study to show the mechanism for hyperlipidemia and hyperglycemia, the interesting things are here. Like say, this combination synergy network of 13 compounds with 23 targets and associated disease conditions, they indicate the hyperglycemic effect here, then hyperlipidemic effect, and in fact, insulin sensitivity network as well. So the combination synergy network or the pharmacology of Trigonella against hyperlipidemia and hyperglycemia proves the mechanistic approach for its used in hyperlipidemia and hyperglycemia when we combine those LCMS-MS profile, in vitro study, in vivo study, and the network pharmacological studies together, we can establish the mechanism of action. And this proves that how the synergy or rather combination synergy helps in establishing the efficacy of the drug in particular context. So like this can be a plant from Ayurveda, this can be a plant from traditional folklore medicines, from Lokasastra Parampara, whatever it may be. Those particular context of establishing or rather validating their therapeutic effect, their phytochemical context chemoprofile is the need of the hour by which we can develop evidences, or rather we can develop more documentations on our own system of medicines, which has a great potentials for drug discovery and drug development. Specifically, so what I believe that this drug development from nature is a traditional medicine inspired approach where we need to give value to both codified, non-codified systems, and there the modern trend on developing the drugs from nature, if we nurture, then we can develop our traditional medicines to a high extent. So in this context, what I want to highlight, one of the things is that we must work on the quality evaluation, validation, chemoprofiling, and related context, which can relate with the mechanistic approach for developing those drugs, and at the end can relate with the chemistry, biology, and therapeutics of the specific components. And here, say, like, we must have to know about the traditional system of medicines and how we can harmonize throughout the country, then ethnopharmacology, ethnomedicines, or the other quality evaluation of this herbal medicinal products, including its qualitative analysis, morphological study, and then knowing the techniques for extractions with the downstream processing, or even bioactive phytocomponents and their analysis, including the thin layer chromatography, high performance thin layer chromatography, or LCMS study, and chemical profiling as such. And at the end, we must have to have those evaluation profile that may be with the bioassay guided isolation, enzyme inhibition study, and even antiviral, antifungal screening, or even the antimicrobial screening of the drug, and leading to the development of specific formulations, including its quality assurance and stability study. With all those, recently we have come up with this document on quality control and evaluation of herbal drugs, where we can evaluate the natural products and traditional medicines based on from farm to pharma. And this is one of the concept we can, this very recently we have come up with this document. And I believe that if we look on those particular contexts, then only we can think about this ethnopharmacology and ethnomedicine in a different context. Now, when we talk about any of these context on chemical profile evaluation, their therapeutic evaluations, and different other things, then and you think about the recent scenario as well, we all are thinking this ethnomedicine, medicinal plant in a very different way. So reimagining ethnopharmacology, and reimagine ethnopharmacology is one of the major aspects on which we have been working, and even there have been the Society for Ethnopharmacology India, or even International Society for Ethnopharmacology as well, in both the place I represent, and where we believe that synergistic approach is one of the major things which need to be. And on that, in fact, to highlight this concept, around 40 countries people we have been able to make, we are having a webinar in every Saturday, alternate Saturday, on this reimagine ethnopharmacology, where we can develop the drugs from our nature with different contexts. And those is available in the society and IBSD website. You can please join. I'll tell you one thing, this Society for Ethnopharmacology was established by with the mentorship of our respected former president, Dr. APJ Abdul Kalam, who really took very good initiative in establishing, or rather guiding us, mentoring us for this, for development of drugs from nature, drugs from our ancestors, and on which we came up with the first issue on special issue on ethnopharmacology in journal, with the special issue on Ayurveda. That was the first landmark in Ayurveda, where a lot of review research articles has been focused, and the first special issue which published on specifically Ayurveda in such a high impact journal. And then we have taken it as a lead on that specific journal again, that on our specific plants resource of the country, and especially issue on ashwagandha. This was initiated in 2019. It is going to be finalized now, and these things is being made. And I believe that with all your support and all, we can come up with so many specialists within this or other journal, which definitely promote our own natural products to a higher extent. And I would like to request you all that you can join with the Society for Ethnopharmacology or International Society for Ethnopharmacology, which can really help you to know more about the things, how the best things are happening, and join in those forum and explore the opportunities as well. Lastly, the friends, healing plants are the nature gifts for everyone. Let us promote those gifts. Let us preserve those gifts for the betterment of the healthcare of the society at large. And definitely, you are welcome to work with IBSD in different forum, or even, as I told, that any of those institutions or organizations, you can link yourself. And please explore the medicinal plants for the therapeutic benefits, specifically documenting them on the scientific evidences, which really can promote our product to a high extent, and we can feel proud that our own natural products can heal the specific disease, which are the nature's gifts for everyone. Thank you so much.
Thank you very much, sir. That really great and a very different and very interesting topic about medicinal plants and how they could be used as a potential source for treating a lot of diseases. So we have a set of questions again by Praveen sir, where he's asking, do you have developed lab for this?
Yeah, definitely. Without lab, how we can work?
No, no, that is true. But so are you doing this all work in your lab? No.
No, we have a lot of collaborations. We have several collaborations in different places, and then with those collaborative work, we are trying to develop something. So, like here, we need, as I told you, a very different group of scientists because say, I am not an Ayurvedacharya, so I do not know anything about Triphala. I was taught about this because I learned some Sanskrit out of my own interest with my Ayurvedic friend. And then I told him that give me some clue by which we can get a polyherbal formulations where maybe two ingredients or one ingredient is similar. That way you identified the marker. Like that, say another 7, 8 formulations we have taken into, and then somewhere say like some place the instrumentation, some place the manpower, and some place the research output, making together and developing the things accordingly.
But if you have the institute, your own institute in-house lab will be the better for this instead of the
Yeah,
collaboration.
definitely we'll try.
Because there will
definitely
be a lot
try
of
that one.
output on that. There is a lot
Yeah.
of work to be done in this area.
Yeah, so we have another question from Tushar. What is combinatorial biocatalysis and combinatorial biosynthesis in drug delivery? Can you explain?
Say we talk about here the combinatorial synergy, combinatorial biocatalysis. That is a synthetic chemistry. We talk here about the combinatorial synergy. Combinatorial synergy means, say like as I told, molecules alone don't is not able to give the same therapeutic benefit what is expected. So here, if the mixtures of the component present therein, we have to explore that one, and this synergy has become now a specific master's course in synergy in different universities abroad. So we have collaborations with several other universities, and by that way we make those things like we have a synergy forum, and there what we are trying to develop based on the synergistic approach of drug development, that this combinatorial chemistry and the synergy together can help us to establish the mechanistic action, and that is the things what we are trying to do.
Okay, thank you so much, sir. It was really
Thank
great.
you.
Yeah,
Thank
now
you.
we'll be moving ahead for the next speaker. So our fourth speaker for the day would be Dr. Soumya Basu, Associate Professor at Cancer and Translational Research Laboratory, Dr. D.Y. Patil Biotechnology and Bioinformatics Institute, Pune. Ma'am, this you can continue now.
Good morning. Am I audible?
Yes, you are perfectly audible.
Thank you. And thanks JAX Foundation for giving me opportunity to talk here. Should I start presenting my screen?
Yes, you can.
Yeah, please just let me know whether it is visible to you.
Yeah, we'll let you know.
Show me. Is it visible?
Yes, it's visible now. Yeah, just make it full screen, ma'am. Yeah, yes, you can continue.
A very good morning to one and all. I am Soumya. I am an associate professor in Dr. D.Y. Patil Biotechnology and Bioinformatics Institute, Pune. Uh, Today, I would like to talk on the cellular immune therapy and with a special emphasis of CAR T cell therapy. Now, I have designed this talk keeping in view that many of the students who will be present here will be grad students or undergrad students. So I wanted to give them cutting edge technology which is taking great care of in patients and in clinical settings. So I have designed this talk in a way that I will first give an overview of this therapy, and later on I will move on the work, what we will do here in Cancer and Translational Research Laboratory, DOI-PVI. So first, I want to give you, I would like to give you the storyline about the different types of cancer therapy, then how chemotherapy, targeted therapy, and immune therapy, they have their own limitations and advantages. Later on, I will move on adaptive cell therapy and then our work here. If we talk about different kinds of cancer therapy, so chemotherapy, hormone therapy, immune therapy, surgery, targeted therapy, radiotherapy, bone marrow transplantations, these are the different ways how a patient is being treated depending on the cancer type, stages, and availability of the therapy. And each of these therapy has their pros and cons as well. If we talk about the chemotherapy, which is the most clinically used therapy in tumor, in cancer, it actually targets rapidly dividing cells. For targeted therapy, proteins which are required for cancer growth and are distinct from normal cells, they can be targeted. And immune therapy, where it uses our own immune system to fight the cancer. Now, none of this therapy is foolproof. They comes with their side effects and limitations like hair loss, nausea, hepatotoxicity, intestinal damage, diarrhea, development of autoimmunity. And sometimes they develop drug resistance. They are not so specific, like chemotherapy is not so specific. And cancer cells, they rapidly become resistant against chemotherapies. They again become resistant to radiotherapy as well. For targeted therapy, it has been observed that developing resistance is very profound. And if we talk about immune therapy, this immune therapy is tailored, personalized, but it comes with a cost that it is very expensive. But if we compare to them, then chemotherapy, as compared to chemotherapy and other therapies, immune therapy show a better effect in clinical settings, though it is very, very expensive. Now, if I move on to the different kinds of immune therapy, this immune therapy can be administered as small molecules, which can modulate the immune system, as well as cell-based therapies. So these cell-based therapies, there are actually 3 or 4 different kinds of cell-based therapies. One is tumor infiltrating lymphocyte therapy or TIL therapy. Another is engineered T cell receptors therapy or TCR therapy. And the third one, which I would like to concentrate more today, is chimeric antigen receptor T cell therapy or CAR T cell therapy. So let's have a look first individually what they are. So this tumor infiltrating lymphocyte therapy or TIL therapy, where doctor or clinician or scientist used to do while they used to give this therapy to the patient. These tumor infiltrating lymphocytes, they are actually harvested from patient's tumor. These TILs, they are encouraged to grow in vitro. They becomes reactive with the immune tumor antigens, and they are being expanded using IL-2, that is interleukin-2. And when there is a sufficient volume of tumor reactive T cells, they are infused into the patient. In case of engineered T cell receptor therapy, here, this is an adaptive T cell therapy, This patient T cells, they have been isolated from the PBMC, that is peripheral blood mononuclear cells. They are isolated, and they are infected with a recombinant virus, and these cells then are allowed to express tumor-specific antigens or tumor-specific chimeric antigens or CARs, and this transduced T cell then expanded in vitro using IL-10 and other cytokine immune interleukins. Again, they are infused into the patient. Now, the third one is chimeric antigen receptor therapy or CAR T cell therapy. This is very interesting. First, we talk about how these CAR T cells are actually works on, how it can be manufactured. These T cells, they are collected from a patient via two techniques. One is leukapheresis or apheresis, and then what this technique used to do, they actually blood is withdrawn from the patient body, and one or more blood components like plasma, platelets, white cells, they are removed. The remaining blood, once we remove the cells, the remaining blood that is returned to the body. Next, we isolate these T cells. These T cells, they are actually re-engineered in the laboratory. Here comes the marvel of molecular biology, RDNA technology in biotechnology applications, where it is currently being used in clinic for patients' benefit and patient survival. It has been saying that CAR T cells, they are actually able to increase patients' life. The T cells, then, which are engineered, they are sent to the laboratory or drug manufacturing facility. You know, in this drug manufacturing facility, they are highly regulated, GMP compliant, and with proper protocols in place, they are then allowed to grow these cells. In these cells, the DNA of these cells have been modified by so that they can produce chimeric antigen receptors on the surface of the cell. And after this re-engineering, the T cells, which are now called chimeric antigen receptor or CAR T cells, they are actually allow the T cells to recognize an antigen on the target tumor cells. These re-engineered T cells then multiplied and expanded by growing the cells in the laboratory. And when there is a sufficient volume or sufficient number of the cells, which is enough to infuse into the patient, then they are actually infused into the patient system. And sometimes few part they have been actually kept cryopreserved. Now, this is a long process, you know. In this long process, it is almost a 21 days process. In this 21 days time, the patient has been kept under chemotherapy so that he will not experience any kind of tumor growth or something like that. Then the patient's body need to be prepared with the help of some kind of chemotherapy so that they will not be able to receive these CAR T cells, and and they will not reject it also. Fine. So in the patient body, these CAR T cells, they actually multiply in a number and they attack the cells. That is, here you can see, they can now recognize these cancer cells. These cancer cells they can recognize, they bind to these cancer cells and they release some molecules which actually reprogram the cancer cells and the cells dies. So this is a marvelous technology designed with the help of biotechnology. Here the innovations comes, but this therapy also is not free of its side effects and also its limitations. So there are several side effects like generation of autoimmunity, generation of other immune system disorders that are there. So patient should be always kept in constant vigilance to rule out those side effects. Now I come to the second part, that in the Cancer Therapeutics Research Laboratory in DYPBI, what we used to do in my laboratory, it has three parts. One, we design the drugs based on the natural products. So natural products, analogs actually we used to design. How we used to design? We screen thousands and thousands of molecules available in the databases with the help of molecular docking and dynamic studies. Then we synthesize the leads generated through this in silico work. So for this synthesis work, I used to take help of my collaborators. And once the lead is generated and characterized properly, then we take this lead to the preclinical validation level. This preclinical validation has two parts. One is first we do in vitro validation using different in vitro methodologies and pathway analysis. And once the lead shows expected result, then we take that lead to the in vivo validation level. So these are some of the publications just came out of our lab. In the first, it is a collaborative work where we have designed some molecules. I would say that is not exactly molecule, that is a nanomaterial. It is not exactly in line of the work what we do here, but it's a collaborative work, and here we have designed the biocompatible material which might be used for orthopedic surgery and replacement thing. Next is here comes the work which is funded by DST, that is the design, synthesis and evaluation of novel structural analogs of natural products as selective modulator of PPR pathway, which is targeted for lung cancer therapy. Here, we have screened more than 20,000 compounds through in silico. And finally, we came up with 5 of the different molecules which we synthesized in lab. And this is the few of the data which shows that our compound, this is actually the lower part is the unpublished data. It shows the EMT transition, role of PPAR gamma, which is modulated. I would not say it is a total agonist, but these are the partial agonists which are able to synthesize, and they are actually able to convert EMT to MET transition. So we know that EMT transition, that is epithelial mesenchymal transition, is a very fundamental stage of metastasis. So our compounds are able to stop metastasis and convert the cellular phenotype from epithelial mesenchymal to mesenchymal epithelial. Few other works which we are currently doing in silico, where we are using artificial intelligence and machine learning approach. This is we have just started a few months back, where it has 2 parts actually, where one we are doing identification of drug target pathways in lung cancer using pathway crosstalk. Now we are developing also some of the machine learning and deep learning tools for designing new T-cell, new epitope for immune therapy design. This project, if we can success, we'll be able to design T-cell neoepitopes, which will be able to understand and recognize cancer antigens more efficiently, so that it can be used kind of T-cell, adaptive T-cell therapies later on when can be translated in vitro. We also work on exosome studies in cancer. So exosomes, you know, they are the small packets which are released from every kind of cell in our body. And these exosomes, they are very much required for the homeostasis, maintaining the homeostasis of our system. And cancer, which as it's a very smart disease, and which is an evolutionary disease. They secrete their own exosomes, and those exosomes, they actually the first molecule which reach to the distant metastatic site and establish the pre-metastatic niche. So one of our project is targeted on the studying these exosomes, modification of these exosomes and exosomal pathways in cancer. So DYPBA is a DSTF supported institute. We have several other infrastructural facilities, including supercomputer, fax, confocal microscopy, tissue culture laboratory, and animal health facility. And this is my group members. So Sangeeta is my PhD student. Gargi, she is working on this artificial intelligence and machine learning project on immune informatics. And on the right side, you are seeing the alumni of few of the alumni of our CTRL lab and the projects they have worked on. And I, of course, without help of the collaborators, it is not possible to work independently. In-house, I work with Dr. Amit, Dr. Rajesh, Priyanka, Sapnil, and my collaboration with our hospital is with Dr. Raut, Hundesar, and Charushi Lagore, and external collaborators with IIRD, BARC, CNCI Kolkata, VIT, and Dr. Rajiv Chikate, he is in Garware College, he is my synthetic collaborators. And my acknowledgement goes to Chancellor, Pro Chancellor, Vice Chancellor, my director, and my research coordinator, and my colleagues, collaborators, and group members without whose support it is not possible to build up any story. And last, not the least, what I want to mention to the younger generation that we need more and more enthusiasm and involvement. There are innovations in biotechnology where without which actually we will not able to sustain, we will not able to fight, not in cancer, but any other thing, any other diseases, any other problems, be it healthcare problems, be it agricultural problems, be it societal problems. So we need young minds to carry out the work. And thank you.
Thank you so much, ma'am. It was really great seeing all the research work at your lab, and it was very inspirational as well. So now we'll be moving ahead. Yeah, just before moving ahead, it's a very kind request for all the speakers to please other with the time, because we are running a bit late. So that's why, in order to just catch up with the time. And now we'll be going ahead with the next speaker for the day, which is Dr. Richa Mahan, professor at Jivaji College, Gwalior. Ma'am, you can start presenting and start your talk, ma'am.
Yes, wait for a minute. I'm sharing my slide.
Ma'am, you're yeah, hello.
Hello, is it audible to all?
Yes, you're audible, ma'am.
Yes,
Yeah.
so please wait for a moment. I'm sharing my slide.
Yeah, okay.
I think something went wrong. Please wait for a moment, sir.
Yeah, no problem, ma'am.
Yeah.
Ma'am, are you able?
Yes. Yes, sir.
Are you
Ye-
able to do it, or is there any problem?
No, no, actually, sir, there is a problem with slide. It is not opening. So I'm trying to open it. It's not working actually.
Maybe you can join from the phone, because even on phone you can
No,
present this.
no, actually, sir, I am joining with this desktop only. Uh,
Okay.
actually, slide is not opening. I don't know what's the problem.
Tanya, are you there?
Excuse me, sir. If you have no problem, can you go ahead with other speaker? Then I'll join.
Okay, fine. I guess we have to do
Yeah,
that. No
yeah,
problem.
yeah. So after
Let
that,
us
I
know
will
if you are
join.
able to do at the end. Maybe, yeah, yeah, okay. So going ahead, our next speaker for the day is Dr. Dharmendra Dubey, sir, assistant professor at Symbiosis International University, Pune. So yeah, sir, you can continue now. Sir, are you there, Dharmendra, sir?
Yes, Aditya.
Yeah,
Yeah. Okay.
yeah, sir.
Yeah.
Yeah, you can continue, sir.
Yeah. Just a few seconds. I hope my slide is visible, Aditya.
Yes, your slide is visible, sir. You can just
Okay.
put it in full
Okay.
screen and start. Yeah.
Okay, so thank you so much, Aditya, and thank you, Jets Foundation, for inviting me. And yesterday I listened at inaugural sessions, Subramaniam sir and Raleigh sir. It was very much informative for me. And so I have so let me introduce myself. I am Dr. Dharmendra Kumar Dubey, assistant professor at Symbiosis Institute of Health Sciences, and I am teaching for hospital healthcare management students, master of public health students, and MSc students here. So I have prepared my slide only focus for the students. Those are no in beginning career for research. So and they need to understand the data set in research. So I'll talk about a little bit about the data set and its analysis. So am I audible? Yeah. Okay.
Yes,
So
yes, you
we
are
will,
audible, sir.
yeah, I will talk here about two things: organizing the data and its measurement. So as we know, in research we organize, we need to understand the research process, which is what is the conceptual phase of the research process, empirical phase of the research process, and analytical phase of the research process, which includes all points: research question, hypothesis, identify the research design, and data collection. And after data collection, data management is very much important, and its analysis and interpretation in appropriate way. So once we need to understand, I hope all the students are aware about the data set, how to collect the data. Data can be collected through the survey with help of the questionnaires, and some organizational through website reporting, direct medical examinations, and direct measurements, and we can collect through that administrative record of the data. So data understanding means, what is data's category means data.
we are collecting data with help of the certain variables. So we need to understand the variables and its presentation. What we generally we do the error when we are going to collect the data and when we are managing the data. So we don't know, generally students generally make a error to it's ordinal data, it's nominal, it's qualitative, it's quantitative. So these points need to understand ki its data is categorical. If categorical, then how will get the information? If data is continuous, which variables will come under the continuous, and it's a discrete, so how we can manage the data in discrete order, and data is binary or dichotomous, like we are taking the getting the information yes or no. So these points is very much important. Otherwise what happen, nowadays this is the modern era, people used to put the data in software, and they run the command and directly they get the value, like your descriptive statistics or inferential statistics. But as a researcher we need to understand whatever software giving the output, is it helping in your results, or you are going to in wrong direction for interpretation. So for that point of view, sometimes what happen in software, read the data in coding format like 0101, and sometimes what happen, people enter the variable information like gender, male represents 0 and female represents 1, or sometimes they used to enter data male represents 1 and female represents 2, and they calculate the measures of central tendency, measures of dispersion like mean, median, mode, and but software calculate 1 plus 2, it gives the average value 1.5. But as a researcher we need to understand, so whatever value we are getting, that is actually representing your data, your outcome, your referring it's your objective or not. So all these points is very important as a beginning career of the researchers. And scale of the data set, if data set is nominal, nominal means if we are getting the marital status like married, unmarried, male, female, is this kind of the data is nominal, but we need to understand it is nominal or not, like once we are collecting the data from different sources. Like here data is ordinal, like if we are taking the level of the disease status, mild, moderate, severe, and our scale is interval or ratio. So all these points we need to understand as a researcher, otherwise we'll get a wrong results, wrong output, and our interpretation would be wrong, and all our whatever our objective, whatever our policy planning, everything will affect. And once we collected all the data, we manage the data with help of the software. So now we need to understand how we will need to create the tables, how can we create the graph, and which graphs is required for which kind of the data set, if data is qualitative or quantitative, and we need the row percentage or column percent, maximum once sometimes I review the research paper, people used to make an error. So sometimes row percentage is required, but they are putting the column percentage, which is not referring your objective. So all these points is very important to once we are analyzing the data. So like identifying distribution of variables, if we have a variables, we need to understand your distributions, means all your possible data is under the your study, or sometimes it makes the outliers which we identify with the error. Sometimes we are taking the entering the data in software like gender 1 and 2, but sometimes we found 3 is there. So we need to identify is 3 is required or not under in terms of our study, our data set. So we need to understand all these points. So like such kind of the data set, so with help of the data set we need to create the tables, means which variable will go in first column, which points will go second column, how we'll calculate the relative frequency, we need total frequency, with help of we can calculate the relative frequency. So all these These are the basic points, but sometimes at a beginning career students make an error, so we need to understand all these points. And sometimes others, like in biostatistics, like age of the patient is given, heart related problems is given, age of the patient is given. So in that constant, how will you create the continuous group, continuous data, and how will put the frequency, how will you calculate the relative frequency. So all these points is required. These are the basic points, everybody knows these, but mechanism, this is the art, how to put the data in front of the audience and how we can interpret it. These are the importance. I hope this calculation all the students can get easily. And how to put your data in terms of the graph. There are, as I shown in previous slides, there are many types of the graphs: histogram, frequency polygon, line diagram, box plots. So which graphs is required in which context, we need to understand all these points. And this is this table seems very simple, but maximum people, researcher, used to make an error. In which column they need to put the number of variables, which column they need to put the frequency, is there row percentage is important or column percentage is important, or they can put the percentage, or they can put the numbers. And if they are going for the descriptive analysis, also they are putting in a tables, how in which way they can put the results in the tables. So mean, standard deviation, if they are using the mean, so standard deviation should be there. If they are using median, interquartile range should be there. So all these points is important. And so as a beginning, so I'm talking in terms of the students, because all these points is referred to the students. So it will be very much useful for the students if they point out the basics things. It's like when we are calculating the measures of central tendency, generally we put the data in software and run the command, will get the mean, median, mode. But we need to understand whatever output we are getting is this measurements is appropriate for such kind of the data set or not. Sometimes qualitative information is given, but people used to calculate the measure of mean, median, modes. Is this appropriate? We need to understand for once we are going to analyze the data. And there are certain quartiles, percentile, quintiles, all are the basic measurements, but it's a very important role in the descriptive statistics analysis and basic interpretation of the basic characteristics of the data. So all these points is very important: range, mean, deviation. This is the measures of dispersion, but it plays very important role in data set. See here, I have put it as some basic points. If see, as we all are know, if there is no variation, there is no statistics. But as a human being, we know, and even a data, everywhere is the variation. So if there is variation, we need to apply the appropriate statistic. See the first plot, there is the all values is equal, so standard deviation would be 0. And if there is something, so we are getting standard deviation. So why I am putting these things? Because sometimes what happen, people are putting the measures of central tendency, they are putting the mean, they are putting the standard deviation, but we need to understand sometimes mean is lower but standard deviation is high. But as we know, as the statistics says, standard deviation always less than the mean. So we need to understand if we are getting the results directly from the software. So if we are getting the standard deviation higher, more than the mean, so we need to understand something is error in our data set. So all these points we need to understand. So there are in statistics, there are certain rules. In normal distribution is one of the key points which adjust the data set. But here I will I have put it some basic points. If we are 60% of the measurements fall within one standard deviation around the mean, this is the empirical rule of the normal distributions, and 95% of the measurements fall within 2 standard deviation around the mean, and 99.7% of the measurements fall within 3 standard deviation around the mean. And at the end, I have put the same thing in with help of one example. This is my last slide. Suppose the reaction time of a particular drug has a normal distribution with a mean of 10 minutes and standard deviation. So here I am going to show the if mean given standard deviation given, so how we'll talk in terms of the data set. Then we can say 68% of the subjects taking the drug will have reaction time between 8 and 12 minutes. And if 95% of the subjects taking the drug, it will have reaction time between 6 to 14 minutes. And if it is going to increase our confidence interval on the with help of the data set, so our reaction time drug will take 4 and 16 minutes. So these are the basic informations. Once students is going to start going to write a research paper, they are collecting the data and going to analyze data and interpreting with help of the referring the objective of the study. So they need to understand all these points, basic things. This is the about my talk. Thank you so much. Aditya, over to you.
Yes, thank you so much, Dharmendra sir. So we have a bit of questions here. So one thing is, what is t-test by Tushar?
So t-test is different measure. So once we are going to know about t-test, chi-square test. So if in terms of the data set, once we have data set, we need to first check the normality condition of the data set. If our data set is following the properties of the normal distribution, then we can apply t-test. Otherwise we'll go for the other non-parametric test. But what t-test, once we have a two groups and we have a mean is given. Suppose for example in a class there are two group of the boys students and girls students, we are getting the weight of the boys students, girls of the girls of the weight of the boys students, weight of the girls students, and we are taking the average weight of the students, average weight of the boys, weight of the boys students and girls students. Now we are going to apply the t-test with help of the t-test following the all the mechanism of the statistical test. We can, so we can, if we are going to reject and accept our hypothesis, with help of that we can say there is difference between boys and girls weight or not. So for mean testing we use the t-test if we have independent sample or dependent sample.
Okay,
Yes.
thank you, sir. So he's also asking about t-test analysis of variance and x-square test.
Yeah, that is x square, that is the, that is known as chi-square test, not a,
Yeah.
that is the notation of the chi-square test. And variance test is analysis of variance test. So once we if we have two groups, then we apply the t-test for mean testing. If we have more than two groups, then we apply analysis of variance one way ANOVA. And chi-square test we use if we have a qualitative data. So we generally use the chi-square test to find the association between the variables. I hope he got the points.
Yeah, yeah. Thank you, sir. So yes, thank you so much.
Okay, Aditya.
Now we'll be moving to the next speaker. But before that, Richa ma'am, is your technical problem solved?
Yes, yes, sir. Yes, sir. Now everything is normal. Can I go ahead,
Yeah,
sir?
you can go ahead. Yeah.
Oh, okay, sir. Okay. So I'm able to present my slide.
Yeah, click on the present now option down
Yeah,
there.
yeah, yeah, yeah. Yes. So it is. Is it visible to you?
Yeah, it's loading. One second. Yes, yes, it's visible. Just make it full screen, ma'am.
Yes, yes.
Down there is an option. Down, down. Yeah, see, down, down. Yeah, yeah, fine. Okay.
Yeah.
Yeah,
Okay.
yeah,
So
ma'am, you
thank.
can continue.
Okay, okay, sir. Thank you so much. And I'm so sorry for the technical issues that was caused from my side. So now everything is okay. And I'm very much thankful to the ZX team that has invited me to present the talk in this such a great platform, and where all the eminent personalities of the all over from India, are gathered here. So basically, first of all, I have to introduce myself. Myself, Dr. Richa Mahant, faculty and training in charge in the Center for Food Technology, Jivaji University, Gwalior, Madhya Pradesh. So, being a biotechnologist, I'm working in different area of biotechnology. Basically, my research interest was uh, disease diagnosis. But I'm also working as an academician. I'm connected with the different UG and PG students. So basically, this talk is I prepared for the students. I think there are a lot of students are connected in this platform. So my topic is the role of biotechnology in the improvement of food quality and nutrition. Is it visible to all now?
Yes,
Hello?
ma'am, it is visible.
Yeah.
Yeah, yeah, you can go
So,
ahead.
so should I go ahead?
Yes, ma'am, you can go
Yeah.
ahead.
Yeah. Okay. Okay. So, as we all very familiar that the biotechnology is the technology which we are now using to change the different type of things. So, if specifically if we have to discuss with related with the food. So the food biotechnology in which we are changing the different things or the different properties of the food material, where we are increasing the nutrition, where we are changing the quality of food, we are working on the yield of the food. So if we go from the past, the biotechnology is used in food production for the thousands of years, not now. It's a very old tradition. So starting with the first loaf of the yeast bread and the first jug of wine, we are that was the technique which we all are familiar with, the fermentation technique. So in modern time, what the things are changing, because as we all are familiar that due to COVID-19 there is a psychological effect in individual and the society. So there are too much change in the lifestyle, and of course there are rapid change in the food habits also. So food consumption and eating habits are significantly impacted because of the new concern due to this COVID-19, the hygiene, safety, and distancing. So that's why now the time has been changed. We have to work on to increase the nutrition of the food. Of course, there are a lot of nutritious foods available, but what happened? We have to increase at the technological level, because now we have to incorporate the hygiene, we have to incorporate the cleanliness in farms also. So that's why now the role of technologies, the role of biotechnologies has been started to cover up these all the things. So now, of course, due to COVID-19, there is also the loss of income earning opportunity. So we have to also work on the increase the crop yield, not only the crop yield, but of course the yield of that processed food also. So basically, I'm working on that food, the processing of the food. Food processing is my work area. So in modern time, what happened? The scientists are seeking ways to alter crops and animal in different ways. We all are very familiar with these all the things. The scientists are developing the resistance to disease, resistant to herbicide, resistant to extreme weather conditions. So different crop varieties are already developed with these aspects. We are now working, the food biotechnologists are working to tasty food also, the processed food which should be tasty, which should be appealing, which should be more demanding in the market. More nutritious food we are preparing, and of course the increasing the crop yields. So the advantage of food biotechnology, of course, we all know that the better overall quality of food we are producing, the increasing the farmers' income, and improvement overall health by the production of nutritious food, and reduction carbon dioxide production. So because what happened, we were using the different type of fungicide, herbicide. So now the technology has given the new opportunity. So we are also working on that area. And if we see all these aspects using this technology, so we all are working on the conserving the natural resources. So impact of biotechnology, basically, if we all know that the genetically modified crops were produced on more than 167 million acres in 18th century in 2003. This is a very old data, 15% increase from 2002. So the US was the largest single producer with more than 60% of the total acreage in production. So these are the some example of genetically modified food. Now, what the biotechnology promises to bring important changes in plant as well as the livestock production. So, in both fields, it will affect all steps of the production chain from agrochemical input and breeding to final and food processing. So, there are two areas where the food technologist working. If we are dealing the animal biotechnology, so they are working on the animal production, and the food biotechnology, so they are basically working on the food technologies. And one of the of course the interrelated area is the agriculture biotechnology. So now the first is the animal, what is the animal production. So application of biotechnology in the animal, in the case of animal, where we are doing the use of biotechnology in animal production has advanced more quickly than its application in plant production. So worldwide, more than one half of all biotechnology research and development expenditure are in the field of human health. All the experimental stage, a large number of drug, diagnostic probes, and vaccine, and so on are frequently applied in livestock production prior to becoming available for use by humans. So development in the pharmaceutical industry therefore have had a considerable ramification for animal production, since many innovation in this area are also applicable to animals. So they also work on the reproduction, the selection, and the breeding. So in the field of reproduction, new biotechniques such as embryo transfer is there, in vitro fertilization is there. I hope all students are very familiar with these technology. So the cloning and the sex determination of embryo have been developed for different type of livestock, for example, in the case of cattle. So this is of considerable interest for breeding program in developing countries, since importing frozen embryos can be less costly than importing live animals. So second one is the animal health. So animal health can be improved with the new biotechnology method of diagnosis and prevention and the control of animal disease. Diagnostic tests based on the use of monoclonal antibodies and new vaccines against viral and bacterial disease are also particularly relevant for the developing countries. So biotechnology research, if we see in this specific the feeding and nutrition area, so in this field the of animal nutrition concentrate on improvement in the enzymatic treatment of food, the addition of inoculant to intelligent, and the fodder for better and faster conversation, and decreasing the anti-nutritional factor in certain plants such as legumes. They are working and which are used as a feed. So in developing countries, such technique might eventually increase the potential range of crops used to feed larger herds of livestock. So experiment with the hormone to improve milk and meat production, such as bovine somatotropin and different recombinant somatotropin has been already produced. So these are the subject of much debate in industrialized countries because of their possible negative effect on animal and farm structure. So in developing countries, however, specific need for increase in productivity can be overriding consideration, which may lead to earlier large scale adoption of this technology, because we all are here discussing about the advances of biotechnology research which we are undergoing on the biotechnology. And of course JEDx has been taken this biotechnology as a topic in this platform. So this technology are doing in this area, of course, the devastating and whatever the disadvantages of using for the using this technology for the feed and the nutrition. So this technology then in many countries, this field represent the fourth area of biotechnology application, the growth and production. So we are also working on the growth and production if we are dealing the animal production. Next is the plant production. So plant production, there is scarcely different aspect of plant production will hot undergo profound change as a result of the application of biotechnology. And commercial application of plant genetic engineering, that is a more relevant with this field, have not yet occurred in the, particularly with the plant production. At the present time, more traditional aspect of biotechnology such as tissue culture have had an important impact, especially in the acceleration of the breeding process for new varieties and in the multiplication of disease-free seed material. So if we discuss about the provision of seed, so there is the plant breeding has been enhanced considerably by in vitro development of improved variety, which are better adapted to a specific environment. So the application of tissue culture has several advantages like the rapid reproduction, multiplication of cultivar, rapid adaptation. And the second one, the reduced use of agrochemicals. So biotechnology can help to reduce the need for agrochemical, which small farmers in developing countries often cannot afford. So a reduction in the use of agrochemical implies fewer residues in the final product. And next is the increased production. So biotechnology can be used in many ways to achieve higher yield. For example, by improving flowering capacity and increasing photosynthesis or the intake of nutrients.
Hello, ma'am. Ma'am,
Yes.
just sorry to interrupt. Actually, we are running late on time. So,
Okay, sir. Okay. No problem, sir. So, and next one is the improving production, improving harvesting. So, the cloning of plant can help to reduce the work necessary for harvesting. And next is the improved storage. So, food shortage would not exist in many countries if the problem of post-harvest losses could be solved. So microbiological reactions by toxicogenic, infective, and spoilage microorganism cause the greatest loss. So biotechnology may contribute to solving these problems also. Next one is the food processing. So this is my working area where we are uh, standardizing different type of technique to increase the food safety, shelf life extension, nutritional value, and texture of food, aroma, flavor in the different type of food using the different agricultural waste substances, developing the taste in during the food processing, and of course the appearance. So basically the fermentation technology we are using to modify such all kind of things, because and what happened we when we are dealing the food processing, so proteins and vitamins are often lost in traditional food processing. So fermentation processes may offer a way to preserve them. So basically we are working on the fermentation technique. So biotechnology can be used for the upgrading of traditional food processing based on the fermentations. And it can also help to eliminate the toxic compound which arise during the food development. And either we can do it by the genetic engineering or through the food processing, or of course there are a lot of technique like the strain improvement of microorganism. So basically what we are doing, we are going through the strain improvement using the genetic engineering or the feedback inhibition during the metabolism. So present day application of biotechnology in the food processing. This actually this is my uh, research topic. So I have to um, especially focus on or make the attention of students on this particular field in the food technology. What there are a lot of scope to work on the food processing. So you can go with the literature which are easily available on the net. So present day application of food biotechnology, especially in the food processing, are far more advanced than application in the field of plant genetic engineering. So the genetic manipulation of microorganisms which are used in the food processing is considerably easier than the manipulation of more complex plants, because if we are modifying the plant, so it takes a lot of time to standardize. Of course the time is also involved here, the time is also the factor here, but if we compare with that the plant changes, so very easily we can go with the food processing. We can incorporate different type of additive to increase the shelf life of product, to increase the food nutritional value on the food. So while the chance to improve food processing is largely neglected when we are going with the plant genetic engineering. So next is the old and new obstacles when you are dealing this the food biotechnology. So obstacles are frequently related with the weak linkage between the international and national research institutes. People are not aware, much more aware regarding this. Poor communication between the national research institution and farmers, and a lack of support measure like the credit schemes and demonstration plot, market outlets, and land holding structure, of course, which dampen the interest of landlord and tenants of in introducing the new technology. So the same barrier that have prevented the acceptance of earlier waves of new technology may also hinder the application of biotechnology, so thereby preventing the realization of its full potential. Furthermore, the rapid increase in the number of technique inventions which constituents property knowledge will make their diffusion in all over the country even more difficult it is. So the next slide is the conclusion. So of course you people have not to wait much time for me. So the conclusion, food biotechnology represents an opportunity for developing countries to realize tangible health and nutritional benefit with sustainable agriculture and new market niches. So in fact, India is actively using this technology, and their support for biotechnology may turn the tide in global public acceptance, because biotechnology has tremendous potential for increasing the food production and improving the food processing, although the real impact will only be felt after a period of time. It will differ from the country to country also. So productivity must first increase in developed country before real benefit can be uh, rapid in developing country. So where biotechnologies, which we are, so applied in the production, destined for domestic market, demonstration effect, and of course stimulate the development in other areas also. In this case, there is considerable scope for cooperation among developing countries. However, where the application of this new technology aims to increase productivity in the export sector, success in some countries could be at the expense of the market position of others also. So in such a these are some things, these are some areas, these are some topics which we have to keep in mind when we are developing any kind of nutrients, and we when we are developing any new technique with related with the food especially. So it's all about my talk. Thank you so much.
Thank you so much, ma'am, for the talk. It was really good. We learned a lot about food technology. So yeah, so now we'll be moving ahead. We don't have any questions as such, but if any question comes in, we'll ask that. So now we'll be moving ahead to the next person, next speaker. So our next speaker for the day is Dr. Bijay Patnaik, scientist C at AIMS Delhi. Sir, you can start presenting.
Yeah, thank you, Aditya. And thank you, Miss Deepa Patil, Miss Tanya Bhagat, and the Jax Foundation for inviting me to present or share my thought in the innovation of biotechnology. So, can I share my slides?
Yes, sir. You can share your slide. Yeah. There is an option down there called present now.
Okay.
Are you able to see that present now option down there?
Yeah. Just a minute.
Yeah.
Give me 2 minutes. I'm unable to share this tab. Okay. Wait. Uh,
Sir, are you able to do?
just a minute. On share screen, Google Chrome. I'm unable to share my screen. Wait. Give me 1 minute. Um, just a minute. Uh, can you see my slide?
No.
Wait a second. Wait. Okay, I got it. Now can you see?
No. Yeah, it's coming now. Yeah. Yes,
Yeah.
we see now.
I'm
Yeah.
so sorry for the delay, for the technical glitch. So, thank you for being patient for my technical glitches and all. So here I would like to share the part of biotechnology that you are dealing with. The title of my topic is Bidomix, a perfect marriage between medical science and biotechnology. By the myself, Bijaranjan Patnaik, I'm working as a scientist at All India Institute of Medical Sciences, New Delhi. So you must know about biotechnology. and you know about biomedical science or medical biotechnology. So after seeing or studied medical biotechnology or biomedical science, why I coined this term, the perfect marriage between medical science and biotechnology. If you see the current pandemic scenario, this COVID scenario, this quick diagnostics, perfect diagnosis, and the non-invasive diagnosis are important tool for the diagnosis of the COVID patient. Those who don't have, those who are like exposed to any COVID patient, whether the patient, the person is having corona or not, for that you need to diagnose. For that, some non-invasive technique or the potential technique should be there in the market to diagnose the corona, the pneumonia. So in the beginning, if you all have gone through these different speakers in the forum, and I don't want to spend more time in the definition of biotechnology. A quick definition is understanding of living organism function at the molecular level. It combines a number of disciplines like biology, physics, chemistry, mathematics, science, and technology. So when biotechnology was fundamental biotechnology was started, it was started from the human when they discovered or they learned to produce beer, bread, and wine. Now the recent advancements are bioprinting. You must heard about 3D printing. You know the biosensors, gene editing, iPSCs. Like recently or last time, Professor Yamanaka got Nobel Prize for induced pluripotent stem cells, and the latest fascinating term that is called CRISPR-Cas technology. So what is biotechnology? It's generally a pool, or it's an ocean of different technique, different tool, different subject, and pool of an ocean of different knowledge like immunology, genetics, plant agriculture, biochemistry, microbiology, fermentation, food technology, and environmental biotechnology. So why we need to innovate more in the biotechnology? So most of the biotechnological products involve at least one biological organism in order to enhance the output of the product or the organism or the any material. We need to develop some intelligence or to process that material for the large scale. So in that process, in the large scale production, the omics comes in the picture. Omics, it's an like it's a collective characterization and quantification of pools of biological molecules that translate into structure, function, and dynamics of an organism. So if you must heard about different omics like genomics, that is the sequencing of DNA, transcriptomics, the sequencing or array of RNA, proteomics like profiling, lipidomics, and metabolomics, everything. So omics is an informal term that refers to field of study in biology with niches as genomics, transcriptomics, and proteomics, like that. And when we utilize all the omics like proteomics, transcriptomics, and genomics to understand the function in the molecular level, that time we can utilize the term called multiomics. When we can sequence DNA, we can sequence the RNA, we can sequence the or we can profile the protein by LCMS, something like that. So in that condition, that is called the understanding, or the study is called multiomics. The holistic intent of omics or the benefits of the omics is to study, to provide an important insight to disease diagnosis and therapy. So as all of you had seen my first slide, that I belongs to All India Institute of Medical Science. My area of interest is respiratory diseases like pulmonary diseases, you can call it lung diseases. So it's a pathological condition affecting in the lung and other parts of the respiratory system like muscles, respiratory system, and all. So conditions are like asthma, COPD, pulmonary fibrosis, pneumonia, sarcoidosis, tuberculosis, and lung cancer. So, and the COVID, if you heard about the severe COVID condition, that is called pneumonia. People used to develop pneumonia in their lungs. So factors contributing to these conditions are like infection, as I mentioned, like pneumonia and tuberculosis are infectious diseases. Tobacco smoking that leads to lung cancer or COPD or pulmonary fibrosis, and different forms of air pollution or genetic studies. So those who have some ancestral disease, they can develop some genetic disease like asthma. It is a genetic disease. You can call it a genetic disease. So respiratory diseases are two types: infectious diseases and non-infectious diseases. And in infectious diseases, tuberculosis comes, and non-infectious diseases, asthma, COPD, lung cancer, pneumonia, and pulmonary fibrosis comes. So what are the diagnoses for this infectious and non-infectious diseases? The infectious disease diagnosis, we go for clinical and physical examinations, lab tests, diagnostic radiology, biopsy. In non-infectious diseases, we go for clinical and physical examinations like infectious diseases, lab test, biopsies, diagnostic radiology, including that PFT is the major standard or the international standard for the diagnosis of asthma and COPD. So if you talk about lab test or the biochemical test like hemoglobin, LDH, and all the biochemical tests used to be done to diagnose the disease, whether the patient is having systematic some inflammation or some condition or not. For that, we need to go for the lab test. For next is pulmonary function test. If you have gone through any pulmonary department, or if you are like accompanying any asthma patient or COPD patient, you might have seen this type of the left hand side picture. You might have seen this kind of picture, this kind of instrument that is called spirometry. That will give you the function of the lung. It will give the lung volume, lung capacity. By the detection of the lung capacity and lung volume, we can say the patient is having asthma or not. So for asthma and COPD, spirometry or the pulmonary function test is the important standard tool for the diagnosis. The next one is the radiological diagnosis. So these are a few images of my few patients. Those had tuberculosis, sarcoidosis, and the last one, the lung cancer. You might have seen the chest X-ray image, but you can't understand what is happening with the lung. You can't even if you see the chest X-ray, you can't diagnose, because we are biotechnology people, we are basic biology people. Second is high resolution computed tomography image, where you can see the lung, some infections are there, but you can't understand that. The last one is PET CT. In that you can see the glowing part. That also you can't understand. It is basically from medical science. Those who are in medical science, they can understand it. And for the disease diagnosis, the invasive bronchoscopy is required, like sarcoidosis, like tuberculosis, like pneumonia. You need to go for bronchoscopy. The first picture you can see lots of doctors are like doing some bronchoscopy. They are putting some endoscope or some scope inside the mouth or nose, and then they go to the bronchi or the trachea or the peripheral lung to see how the growth is there, what type of growth is there, and they just used to collect the sample from the image. You can see the middle image, that abnormal growth. You can see some abnormalities. The doctor, the expert, used to collect the sample from that abnormal growth, and they send it for histopathological analysis. The last image is the histopathological images, where you can see lots of inflammation. Here also, for the diagnosis, you have to go for invasive technique. Here in India, all of you know that we are not that much good in diagnosis or medical science, or the facility of Indian culture is not that much good. So you need to be expert, or the medical expert team should be there to collect the sample to analyze the sample and the definitive diagnosis of the particular disease. And so why we need innovation in the diagnosis? For proper identification of the correct pathological type of respiratory disease, like as I said, sarcoidosis and tuberculosis. You can, if you see the image or X-ray or CT, you can't differentiate between sarcoidosis and tuberculosis. Even doctors sometimes fail to detect the patient is having sarcoidosis or tuberculosis. Even in case of lung cancer also, if the patient is not having metastasis or FNAC was negative, in that condition, the patient is, the doctor sometimes fail to diagnose the proper condition of the lung. For that, we need some innovation in the diagnosis. So in the second is blood biomarkers. Previously, my previous speakers have mentioned about markers like RNA, LNC, RNA, and protein biomarkers. So in case of blood biomarkers, we should have the knowledge of their specificity, sensitivity, and the release of different cytokines or different molecules from different type of cells. If we collect the blood sample from the patient, that will come from different part of the vital organs. So if the patient is having lung cancer, if we collect the blood sample from the veins, that you may miss some conditions, some cytokines in the blood biomarker. The third problem is the current imaging modalities are limited in their sensitivity and specificity for the early detection or the definitive diagnosis of the condition. And the invasive examinations remain gold standard. Till now, we are rely on the invasive diagnosis, as I had shown in my previous slide, that doctors are missed when they undergo any patient and they used to diagnose any patient having tuberculosis or sarcoidosis. They missed. Even in case of lung cancer also, if the patient is even tuberculosis, the doctor will diagnose that patient with lung cancer and give the alternative therapy like ATT or the anti-tuberculosis treatment. If the patient is having tuberculosis and the doctor is diagnosed that patient with lung cancer, they will go for chemotherapy and the radiation therapy. So in that condition, the patient condition may be deteriorated. For these reasons, we need some early diagnosis or any invasive diagnosis by which we can do some definitive diagnosis of the disease. So in my previous presentation, like Dr. Lopamudra also mentioned, early diagnosis of breast cancer must be there to diagnose the breast cancer in the early stage, so that we can prevent the progression of the disease. And it will help if we diagnose the disease in the early stage. We can go for the treatment. Even we can go for the better outcome of the treatment. So here the breathomics come. So breathomics, if you breathe out, like you can feel that lot of water droplets come from your mouth, air come out, and sometimes your friend may say you that okay, your mouth is like smelling so bad, or the pungent smell coming from your mouth. So if we detect that thing directly from the patient is having lung cancer, if we with some technique, some tool, if we detect that thing, whether the patient is having lung cancer or TB or something like that, that will be the beneficial or the early diagnosis of the disease. So in breathomics, we go for two different approaches: direct analysis, and the second one is storage. So in direct analysis, like PFT, radiology, like that, we directly see or they visualize the clear picture of the lung. But the sensitivity and specificity sometimes we can face. But if we analyze the breath, if we detect some disease, some condition in the breath, that would be beneficial for the treatment. And if we store the sample, like in biomedical sciences and medical biotechnology, you must be heard about collecting blood sample, lava sample, tissue sample, and we go for the cell isolation, then RNA isolation or protein isolation. Then we process that sample, and we come with the conclusion that the patient is having this protein and this patient is this RNA. So that is indirect analysis. We store the sample and go for the analysis. So breathomics, as I had mentioned, that every breath that we exhale out contains hundreds of different molecules like droplets. In droplets, we can see we can analyze some new molecule. This fingerprint of breath, as I mentioned, if you feel a pungent smell or some bad smell from your friend's mouth, you will call it your mouth is like smelling very bad. So in that, if you print that breath, we can give some information. We can collect some information of the condition or the disease of the lung. It was breathomics started from ancient Greek, like from ancient Chinese medical in medicine. There in the ancient Greek or the ancient Chinese, they used to smell the sweat condition, sweet breath, and they used to diagnose the patient is having diabetes or the sugar or not. So the breathomics started from the ancient Greek or the ancient Chinese society. So come to the breathomics. If you see the picture, there are two major parts of the lung. The first one is the trachea coming down, then two different parts or two different like part of the lung. You can see if you focus of that lung, you can see a small branches, some cauliflower like thing that are called alveoli. So if in the bigger picture you can see some gaps are there. In that gap, air is there, liquid is there. So if we collect the breath from that part, from the alveoli, from the bronchi, from the trachea, and go for the analysis, in that condition, that would call breathomics. In breathomics, it is two different parts. One is analysis of breath, the air by gas chromatography or the electronic nose, and the second one is the analysis of metabolites like cytokine or proteins or something like that. So the breathomics can be called as a non-invasive technique or a potential technique to detect the disease condition. So as I had mentioned in my previous slide, the volatile organic compounds present in the air. The soluble volatile organic compound will be present in the water droplet, and the non-soluble volatile organic compound will be there in the air. So volatile organic compounds are the chemicals which have high vapor pressure at room temperature. So in exhale breath, a lot of volatile organic compounds coming out from your mouth like acetone, methanol, ethanol, propanol, isoprene. So if you have studied how they smell, how the odor comes from any garbage or anything, it basically depends on the VOC, the volatile organic compound. So based on the odor, the electronic nose, which is a artificial sensor, that detect the presence of volatile organic compound in the breath air. So recent advances in the odor sensing technology that will give the signal of particular VOC or the particular group of compounds in the electronic nose, and that is called the advancement or the innovation of the biotechnology. So it rely on the area of vapor sensor that respond to specific stereochemical characteristics. As I had mentioned in my previous slide, that biotechnology is a pool of knowledge where biochemistry is also there. So this is also a part of biochemistry. Like the human nose, the electronic nose plays the counterpart role to sense the smell. In different memes, you might have seen the left-hand side picture, like the world worst job or the worst job in the world. So they used to inhale the body odor, and they used to detect some disease or some for perfume generation, for the perfume preparation. They used to do this kind of test. And interestingly, dogs can be used for the disease diagnosis. All of you know that dogs have very good olfactory receptors. the dog squad is there for that for like for the following any criminal like that. So previous like in 2006 there was a paper published. In that they trained some dogs in lung cancer and breast cancer. After that they send those dogs to different patient, unknown patients, those who had some clinical features, and the dogs diagnosed the breast cancer, the presence of breast cancer and the lung cancer in those patients. Later on those patients were diagnosed with the lung cancer and breast cancer. Published the research article says that the dogs can detect lung cancer and breast cancer in
Hello, sir.
97% of accuracy. Yeah.
Sir, sorry to interrupt. Actually, the time limit is actually done. Can you
Just
please?
keep it 2 minutes. Yeah, I have only 2-3 slides more. So concept of electronic nose is it will sense the smell. It is like acquisition in the brain or the sensor of the nose, process it and give the what type of VOC is present in the sample. So this is the electronic nose, and the right hand side image you can see the how VOC can be detected. The spike you can see the blue spike, the green spike, and the red spike. These are the VOCs can be detected in the breath of the patient. This is one of my asthmatic patients VOCs. So the next one is the axial breath condensate. As I had mentioned in the earlier slide that the breath out you can feel some droplets, water droplets. It contains different molecules. It's coming out from the airway lining. Okay, so the axial breath mainly contains, people have like detected different cytokines, proteins, DNA, and RNA. Surprisingly, those EBC of the patient like were detected with different microsatellite DNA. So in EBC we can check the pH of the lung, the respiratory, or the axial breath condensate, or we can check the different redox potential, and we can check different cytokines as I had mentioned here, different microRNA, small RNA, like that. And previously one of my previous speakers had mentioned that microRNAs can be used for biomarkers, potential biomarkers. So basically axial breath condensate is a the breath coming out from the airway lining can be collected, can be processed, and from that process sample we can go for analysis of protein, cytokine, RNA, and microRNA. Basically we are focusing on small RNAs and cytokines in different respiratory disease patients. So this is a slide of recently published paper. People have developed a human proteomics database. Here you can check, you can go for any particular disease and you can check whether the patient is having any VOC or not. So cumulatively, or we can say the breathomics is the pool of genomics, transcriptomics, metabolomics, and proteomics. And if we analyze all the things, breathomics will come and we can directly diagnose the disease. The patient is having lung cancer or some respiratory disease or not. We can detect it directly. So we don't have to keep the sample. We can process the sample directly and tell that patient is having some respiratory conditions or not. So in future directions, you must have seen the pollution of Delhi, and this is the picture of polluted polluted air. The lung, you can see the image of lung in the pollution. We can check the breathomics of those healthy individuals who are living in Delhi. We can say that the patient, the human being, the healthy control is having any condition in their lungs or not. In case of pneumonia also, we can go for the breathomics. These are our future directions. This is our team, head of our department, Dr. Anant Mohan, the director of AIMS, Dr. Ranjit Guleria, and director of IGIB, Dr. Anurag Agrawal. And this is an ICMR funded project where we are working on breathomics, or we are applying science in the breath to analyze whether the patient can be detected non-invasively directly or not. So thank you. And last not the least, I would like to thank Dr. Jyoti Mohi H, who is my guru, my teacher, who taught me what is science and what is research. And because of her, I could learn how to do research. And she taught me from the pipetting and the handling any research program. Thank you.
Yeah, thank you so much, sir. It was really great listening to all about bathomics and the unique relation between it. So we have one question here by Jitender sir and Parul. If two people are perfectly healthy, will their breath print unique to them like the fingerprint, or will be same for everyone?
No, it won't be same for everyone because every person has their own smell, own breath smell. So smell comes from VOC, volatile organic compounds. So we can't say that patient, though healthy individuals have similar pattern, but we can say if we compare between asthma or any respiratory disease with the healthy control, so we can say that we have found some difference in asthma and COPD patient in comparison with healthy control.
Okay. And one more question is, what is okay, breath metabolites are highly dynamic and would strongly depend on food habits and the last diet taken. How do you deal with that? Also, what is the typical concentration of metabolites seen in the breath? By Jitender.
Yeah, but like the food habit, in case of food habit, that is why we are collecting the sample from different parts of the country so that we can, like, South Indian food habit is different from our North Indian food habit or East Indian food habit or West Indian food habit. So in healthy controls, you can't see that much difference in the VOC. Okay, so if we compare with the any disease condition, some differences will be there, but we won't find we won't find any large differences between the food in case of food habits. And in case of concentration, yeah, we can calculate. You can do for we can go for the GCMS where we can say we can measure the concentration of the VOC. Yeah, we can do that.
Okay. Thank you so much, sir, for the amazing talk. Now we'll be going ahead. So you can stop presenting the screen.
Yeah, sure.
Yeah. So now our final speaker for today is Dr. K. Subramanian, professor at Banari Amman University of Institute of Technology, Tamil Nadu. So sir, you can start presenting the screen.
Okay, thank you very much. First of all, I thank the Jai Foundation for inviting me to give a lecture in the National Biotech Summit. The slides are visible?
Yes, yes, it is visible,
Hello.
sir. Yeah, it is visible, sir.
Hello.
Hello. Yeah, sir, it's visible.
Visible.
Yeah.
So shall I go ahead?
Yeah, you can go ahead. Just make the screen full screen and then we can go ahead. Yeah. Yes, sir, you can go ahead.
Yeah. So this is my topic is mainly on the biodegradable polymer biomaterials by biomedical applications. So mainly it will be focused on it is student oriented. So first I will present some of the basics on biopolymers, followed by a brief review on the biomaterials that is used in the medical applications. Right. So polymers are the organic molecules, giant molecules that are formed by joining these simple molecules by appropriate chemical reaction known as polymerization. Right. Much of the comfort that we are enjoying today are attributed to the developments in polymer science. Right. So polymer plays a significant role in every walks of life. It is very difficult to imagine life without polymers. That is, the food we eat, the cloth we wear, are all polymers. Right. About nearly more than 400 million tons of the polymers are produced annually, and it contributes a lot for the economic growth. This is a multi-billion business field, right? So nearly about polymers, generally we can classify polymers as natural and synthetic. The synthetic polymers are derived from the petroleum resources. Nearly 4% of the total crude oil that is produced are used for making these polymers. And out of the synthetic polymer derived from petroleum, 35 to 45% are used for packaging applications, and these materials only cause pollution because the synthetic polymers derived from petroleum resources are not degradable. The major cause of the pollution is attributed to the improper handling of these packaged materials when it is disposed literally, that is here and there. So if it is used properly, that is, if it three R concept is followed, that is reduce, reuse, and recycle, so we can minimize the problems that are faced by the disposing of this synthetic polymer waste, right? But however, we cannot imagine life without polymers. Much of the comfort what we are enjoying today are attributed to the developments of the polymer science. The entire body is made up of polymers. Most of the organic materials present on the earth are polymeric in nature, right? For example, the proteins and nucleic acids, the polysaccharides, glycoproteins, antibodies, antigens, all of them are polymers in nature. So the biopolymers are produced from plant, animals, bacteria. They can also synthesize in laboratory using the biotechnology. They can be synthesized by chemical route also. So now I will focus only on the biopolymers. First, I will give you a brief overview of the biopolymers for the benefit of the students, because this may ignite some new ideas in the minds which we promote research in the multi-billion-dollar applied chemistry. That is a polymer is a branch of chemistry. It is application oriented. So biopolymers have many applications. So they are formed during the life cycle of green plants, animals, bacteria, and fungi. About 30% by weight of the human body is made up of that is collagen. Collagen is a polypeptide where the three polypeptide chains are intertwined, forming a helical structure, triplex helix. So it has biopolymers have many applications in the pharmaceutical and medical fields. So biopolymers, that is, if you want to use a biopolymer for medical applications, this should possess some specific properties. That is, it should be biocompatible, biodegradable to non-toxic property, low antigenicity or non-antigenicity, high bioactivity, processability to complicated shapes with appropriate porosity, the ability to support cell growth, proliferation, should possess appropriate mechanical properties, and we should also maintain the mechanical property till its service time. So the properties of biopolymers would be renewable, sustainable, biodegradable, non-toxic, non-immunogenic, non-carcinogenic, non-thrombogenic, and carbon neutral. These properties are not associated with the synthetic polymer derived from petroleum resources. For medical applications, the polymers would possess all these features. So normally the biopolymers are broken down into biologically acceptable molecules which can be metabolized and removed useful metabolic pathway without leaving any toxic residue in the body. Right? These are, for example, unlike the synthetic polymer derived petroleum resources, when these biopolymers are exposed, that is disposed in the land with composting, resulting in formation of carbon dioxide, water, and reuse. So it plays a significant role in circular economy. Right? For example, this carbon dioxide water plant can absorb and synthesize carbohydrates. The carbohydrates can be saccharified to glucose. Glucose is fermented to give lactic acid. Lactic acid can be converted to polylactate. This is a biopolymer. A bacteria consumes glucose and form polyhydroxyalkanate. This is also a biopolymer, right? So if all the plastics in the world are replaced by bioplastics, and the energy used in the process came from the renewable resources, about 4% of the fossil fuel can be that is saved, or we can bring down the consumption of the fossil by about 4% because of the following features associated with biopolymers, that is low energy requirement, low carbon dioxide emission, low greenhouse gas emission, low water consumption, because water is generated and hence it requires only less water compared to the conventional synthetic polymers. So we can classify polymers into natural and synthetic. Again, biodegradable. Biodegradable polymer again can classified into synthetic biodegradable, natural biodegradable. Synthetic biodegradable, for example, polyaliphatic polyesters, polyanhydride, polyphosphazine, polyamino acids. For example, polylysine, polyaspartic acid, polymalic acid, polysabbasic anhydride. These are all the synthetic biodegradable polymers. Natural biodegradable polymers are plenty: albumin, collagen, textile, gelatin, pectin, starch, carrageenan, xylem, gomarabic, guargam, xanthan, chitosan, hyaluronic acid, soya protein, whey protein, casein, cellulose, bacterial cellulose, plant cellulose, glulan, etc. These are some of the examples of the natural biodegradable polymers. So based on the application, again, these polymers can also be classified based on the application, because based on the application, we can classify biodegradable polymers as any kind of polymer, as a thermoplastic or thermosetting plastic, fiber, elastomer, adhesives, sealant, cement. Depending upon the applications, these polymers can be classified under these categories. So some of the examples of biopolymers I listed out: polyester, for example, polyhydroxyalquanone, polylactic acid, polycaprolactone. These are the polyesters. Protein, silk, collagen, wool, elastin, resilient, polyamino acid, polyglycine, soya protein, zinc, wheat gluten, casein, serum albumin. These are the protein biopolymers. Polysaccharide, for example, bacterial polysaccharide, xanthan, textan, gelatin, levain, cardolin, polygalactosamine, cellulose. These are the bacterial polysaccharides. Fungal polysaccharides, for example, glulan, leucinin, yeast glucans. These are the fungal polysaccharides. Plant or algal polysaccharides: starch, cellulose, agar, alginate, carrageenan, pectin. These are polysaccharides of plant and algal origins. Polysaccharide, animal origin: chitin, hyaluronic acid, glycogen. These are the polysaccharides of animal origins. So lipids, surfactants, of course, these are also, some of the lipids can be associated with the protein, for example, that is proteolipids, glycolipids. These are also polymers. Polyphenols, lignin is also a polymer derived from plant resources. Apart from that, there is also crystalline form shellac, polygamma glutamic acid. Natural rubber, this is also a polymer synthesis by the rubber tree. It is a hydrocarbon polymer, right? So synthetic polymers can also be made from the fats and oil, because the fatty acids and the glycerol can be combined together, forming complex polyesters which are of biodegradable nature. So one of the important properties of the polymers is because we can fine-tune the properties of polymers for various applications. That is easy with the polymers and is difficult with the metals. So polymers have unlike simple molecules, molecular weight of the polymers are not same. It is variable, because if you take polyethylene, you can have any molecular weight. If you take protein, you can have any molecular weight. So they will have polydispersing index, because they will have molecular weight distribution, and hence the polydispersing index and the molecular weight. These are the two parameters which will decide the mechanical properties and performance features of the biopolymers. And glass transition temperature, that is the temperature above which a polymer becomes flexible, below which it becomes rigid. And the processability of a biopolymer is decided with the glass transition temperature. You can play with these parameters by appropriate modification of the structure, by appropriate choice of the raw material that has been chosen for the polymerization, as well as by fine-tuning the structure of the polymer, right? So degradability, biodegradability, photodegradability, thermodegradability, thermooxidative degradability, mechanochemical degradability. These are the possible ways by means of which the polymer may degrade. You can fine-tune the degradability, rate of degradations, etc., by introducing appropriate weak links in the polymer. This can be done by modifying the existing natural polymers, or by introducing the weak links during synthesis of the synthetic biodegradable polymers. So another is the mechanical property, that is plays a significant role, which you can alter it by introducing or fine-tuning the structure, right? Next is the crystalline nature, amorphous features. These you can play with by processability, by their mode of polymerization mechanism. You can play with this parameter, because highly crystalline polymers are rigid in nature. tensile strength will be more. Amorphous polymer will become flexible. Hence, you can play with these things either during the polymer synthesis or by modifying the polymer or by processing the polymer. So you can modify the biopolymers by various ways, by blending, by interpreting network formation, grafting, copolymerizing, cross-linking, derivatization, introducing the function group, introduce hydrophilic moieties, hydrophobic moieties, etc. You can change the solubility, gellability, and swellability, all these things. All these parameters can be easily fine-tuned by appropriate structural modifications or by mixing. So that is the secondly, they're all the polymers are non-corrosive. Unlike metals, the polymers are non-corrosive. These are the special features associated with the polymers, unlike metals, and hence they are the preferred material for biomedical application. But the only drawback is the poor mechanical properties. Right? Polymers do not generate microwaves or electrolytic current, unlike metals when it is implanted in the body. They are corrosion resistant. So polymers are organic in nature. The majority of their biomolecules present are organic. Hence, there is a better compatibility. Where metal is inorganic, they are not biocompatible. They can easily microscopically evaluated. They are more aesthetically pleasing, but the disadvantage is the poor mechanical properties. However, this can improve by appropriate modification technique by fine-tuning the structure by the selection of the monomers that has been chosen. So what are the various parameters that will affect the biodegradability of polymer? There are morphological features, chemical factors, physical factors. Morphological factors means shape, size, architecture, crystallinity, amorphous features, chemical structure, types of the linkage, whether it's glycosidic, amide linkage, peroxide linkage, or ester linkage, or ether linkage. So this type of linkage also can decide the stability, biodegradability, and the composition, and the configuration, presence of ionic groups, cross-links, molecular weight distribution, presence of low molecular weight compounds. So these are the various chemical parameters that will contribute for the biostability or biodegradability. Apart from the physical factors like sterilization process, because whenever we introduce a polymer inside the body, it has to be sterilized. So whether we are using what type of sterilization, whether we use a steam sterilization, or we are using epoxy sterilization, or use a radiation sterilization, all these factors will also decide the stability and also the processing conditions. So the different applications of biopolymers, they can be used as coatings, making fibers, plastics, adhesives, sealants, cosmetics. These used in oil industry, paper industry, textile industry for sizing. For example, starch is used for sizing, for making cloths, for making fibers, for making biodegradable cloth. In water treatment, biomedical applications, pharmaceutical applications, automotive applications, for making rubber, elastomer, which is used in the automotive industry, and for packaging applications. Because this packaging application, we have 35 to 40% of the synthetic polymers are usually contribute a lot for the pollutions. Hence, if you are able to replace these polymers by the biodegradable polymers, we may be able to solve the pollution problem created by the synthetic polymers for these applications. The global biopolymer can also be classified based on the market potential. So they will be categorized as automotive, agriculture, consumer products, bottles, textiles, packaging, medical, and other applications. So what are the typical medical applications of biopolymer? It is used for scaffold for tissue engineering, regenerative medicines, wound dressings, bandages, transdermal patches, blood bag, catheters, dental cements, bone cement, bioadhesive in surgery. So all these things are based on the biopolymer. You can formulate all these things using biopolymers. And dentistry, dental cement, stents, sutures, drug carriers, and drug delivery. This is a very promising area where many polymers are being used and modified for specific drug delivery applications. It's a supportive body as implants, contact lens, intraocular lens. All these are made up of the polymers, coatings, disposable personal hygienic personal diapers. Right, so majority of the diaper polymers use superabsorbent polymers in the diaper applications are polymers that derived synthetic polymers, petro-derived. If so, there is ample scope to
Sir?
make. Yes.
Sir, sorry to
Yes.
interrupt. Yeah, actually,
Hello.
Hello, sir. There is a bit time constraint there. Actually.
Oh, okay. I will go through this quickly. Go through this one. So, advantage of biopolymer drug carriers: targeted delivery, sustained stabilization, decrease in dose frequency, minimize side effects, improve patient compliance, control delivery by control degradations. So these are some of the biomedical applications of the conventional polymers: silicon Teflon, diagram, biorethanes for catheter, vascular catheters, ophthalmic applications. These are some of the other applications of the petroderived polymers in medicines. Then these are some of the structures of the amino cellulose, polyhydroxybutyrate, polylactic acid, polybutyl succinate, polycaprolactone, aliphatic aromatic resins, carboxymethyl cellulose. This is the structure of the structure. It's a collagen, right? It's a polypeptide, so 30% weight of the body contains a collagen. This is used for the this collagen is used for drug delivery as a carrier, and also it is in the tissue engineering for making scaffolds, etc. And also it is collagens polyps are used in the management of severe burns, the skin replacement. Where the collagen based is used in the tissue engineering. It's for wound, that is for making batches for wounds. Collagen nanoparticles also used for in the drug delivery applications. The collagens is capable of stopping bleeding, so we can make a collagen film in the power to start bleeding. It's also used. Okay, I will quickly go through the chitosan. This is this is all the marine polysaccharides. So chitosan hydroxyapatite is a mixed forming scaffold in bone tissue engineering. It is the second most abundant polymer after cellulose in the earth. And heavy cellulose is the third most abundant polymer on the earth. There is also polysaccharides. The chitosan bioactivity, biodegradability, selective permeability, polyelectrolyte action, antimicrobial activity, ability to form gel and film, etc., etc. They can also form chelations with many metal ions, so they can be used to remove the poisonous metals in the intestine tract. So chitosan, this is the chitin, derived from the marine species, crab, etc. If you remove these, if you convert the unstable amino group, it becomes a chitosan. It is a polyglucosamine. So cellulose, xylem, these are all the polysaccharides. This is structure of cellulose, xylem. It is a heteropolysaccharides. So majority of the plant derived polysaccharides are made up of galactose, glucose, and mannose, or their derivatives. So mannonic acid, glucuronic acid, etc. So arginate, it is the algal polysaccharides, right? It is used in enzyme emulation as a carrier in controlled drug delivery, in scaffold fabrication, or tissue engineering, regenerative medicine. So in the wound dressing, this is a carrageenan. This is also a high molecular polysaccharide. Locus, derived from locus. That is, sorry, it contains, this is also heteropolysaccharide. In fact, this carrageenan inhibit many viruses. For example, carrageenan is a selective inhibitor of many enveloped virus. For example, HSV virus, HIV virus, even the COVID-19, which is also enveloped virus. This carrageenan can also inhibit the COVID-19. So it's also a good substitute for gelatin in the hard capsules. So gum arabic, xanthan gum, these are the important polysaccharide polymers derived from the plant resources. Right. So pectin is also used for mucoid batches with carbapole and chitosan. Pectin is also used to remove lead and mercury from the respiratory organs and the gastrointestinal tract. The appropriate formulation. This is the structure. This hyaluronic acid is a heteropolysaccharide. It's also biodegradable. Used in medical substitution replacement of eye surgery and as a carrier for ophthalmic drug delivery, used in wound healing by extracellular regeneration, topical treatment for dry eye syndrome, intra-articular injection as a filler rejuvenative medicine for wrinkles. Yesterday, is used to prevent bacterial adhesion to dental implants, intraocular lenses, catheters, etc. So one of the important biopolymers, polylactic acid, which is obtained from the corn or any starch, by saccharification. After the glucose is fermented to lactic acid, the lactic acid converted to lactide by heating, and the lactide is substituted ring opening polymerization to form polylactate, which can be converted to fiber by spinning, by extrusion in the form of film, thermorefund, bottle, woven, non-woven. So you can make an appropriate product out of these polymers by appropriate molding techniques, maybe injection molding or resin transfer molding or compression molding or blow molding. So these are the different molding techniques where you can fabricate the different types of products for the application requirements from the biopolymers, either extracted from the plant resources or synthesis in the laboratory, or to by appropriate biotechnology process. Right. So this is the how the polylactic acid is prepared in the laboratory. Right. Is a so PLA is find extensive use for sutures, pins, dialysis membranes, and drug delivery devices. It's also employed in preparations for the bioplastic for various applications. Right, this is the polyhydroxyalkanate produced from bacteria. So depending upon the N value, you can have a medium chain length or short chain length. So depending upon the nutrient that you are taking glucose or fatty acids, you can play with the architecture of the chemical structure of the polymer that is synthesized in the bacteria. So the majority of these the polyhydroxyalkanate can be used in the food industry.
Sir, sorry to interrupt, but the time is crossed.
Okay, right. So I have not come into the biomaterials, right? Anyhow, so biomaterial is defined as any systematically, pharmacologically unit substance or combination of substance or synthetic material utilized for the implantation within or incorporation in the living system to augment or replace functions of living tissues, organs of the body as a whole, or part for any period of time to improve the quality of life or save the life of patients, right? So, so the biomaterial can be maybe a polymer base or metal base or semiconductor base or ceramic base or composite. But here I will focus only on the polymer based biomaterials. So this is when the biomaterial is implanted in the body, it is the interface. So there is biomaterial should not absorb or elicit unwanted reactions with the biomolecules when it come in contact with the interface. This is the biofluids or the tissue. This is the biomaterial. This is the interface. So these are the various parts of the body where you can make a
Sir, you're not audible. Sir, you're not audible. Hello. One second. Yeah, I guess sir has some technical problems. So if there are any questions for sir, they can put up in the chat box. We can answer them when he's back, once he's back online. So I guess till then I'll proceed ahead then now. One second. Yeah. So till then, so that's one announcement I would like to make here. So we would like I would like to explain you about the JAX membership and its benefit programs. So they have we have two membership programs. One is the youth membership and one is the advisory membership, which youth is for the professional, youth is for the students, and advisory membership is for the professionals. There are a lot of benefits and opportunities which you would gain by being a part of this member of this organization, which are like you would like to be a part of international summit programs by BRICS, United Nations and other bodies. You will get an invitation. You will be also part of a lot of national events like this one. This is a biotechnology summit. There will be more summits coming up in the future by Jack's Foundation. Your organizational skill will also get improved. Networking with all kinds of corporate and institutions, and internship program especially is a highlight for the students there, and national as well as abroad scholarship and exchange programs are also there. They also offer abroad studies based on the performance during the year. So any kind of queries or questions, you can visit the Jax Foundation website and then register as a member and solve more queries there. So now I would like to hand over the today's proceedings to Miss Moini. Ma'am, are you there?
Yes, Aditya, I'm there.
Yeah.
So we have to wait for sir, or shall we proceed?
I guess sir is back now. So
Well, there's a dealing, I think it is. Okay. So thank you very much.
Yeah, thank you,
If
sir.
there are any questions, you can take
Yeah,
them
one
first.
second. There's one question by Hariom here. How do we find the transition temperature of any substance, and in particular of glass, and how phases describe these properties?
So constant temperature, glass constant temperature can be determined using thermal analysis, differential scanning calorimetry, and a differential, that is DSC technique. You can find out, or thermomechanical analysis. These are the two analytical techniques widely employed to determine the glass constant temperature. So the polymer may be 100% crystalline. It will show only a melting point. If it is a semi-crystalline, it will show glass constant as well as a melting point.
Okay, sir. Yeah, thank you so much, sir. It was very great. So I would like to thank all the speakers, first of all, for day two as well for day one for successfully, we have completed this whole summit for 2 days. And as I said yesterday, it was quite a different range of topics as well. If you would see, no topic is overlapping with the other. You see the diversity here. So that is, I really like the whole thing. So yeah, moving ahead. Yes, ma'am, you can take over. Mohini, ma'am. Yeah.
Thank you, Aditya, for beautifully moderating, and all the eminent speakers for delivering such knowledgeable and interesting talk today. Now we will start with a group discussion round, and in this round, all our student scholars are there to participate. So I'm calling the name of all the participants. Yamini Purohit. So please say yes if you are present. Yamini Purohit. Is she there? Neha Bhandari. Our student scholars are not yet joined. Srishti Pandey. Am I audible?
Yes, ma'am,
Someone.
you are audible. Yeah, you are audible. I guess those three have not joined. You can continue ahead with the participants and maybe they'll come, I guess.
Okay, Srishti Pandey. Janani Saptarshi.
Yes.
Okay, Sumiran Singh.
Yes, ma'am.
Ayushi Bhanushali. Julie Kale.
Yes, ma'am.
Priyanshi Kaushal.
Present, ma'am.
Ayushi Sengupta.
Yes, ma'am.
And Aditya Acharya. Yeah, he is
Yes,
there.
ma'am, I am there, yeah.
So now I tell the rules for today's group discussion. Every one of you will get 2 minutes of time to speak over the topic. Your topic of today is, does biotechnology development help in overall development of country? And the participant will must speak only when their name has been called. Also, I would like to invite and request all our speakers present here to give their viewpoints on the student discussion. So all sirs and ma'ams are requested to be present here for guiding them. So now we will start with our first participant. I think Somiran Singh is present. So
Yes, ma'am.
so you can start speaking. And your time is of 2 minutes. So please be on your time.
Okay,
You can
ma'am.
start.
So thank you very much for giving me this opportunity. So today's topic is that is biotechnology very much efficient for the development of a country? And I would say yes, sure it is. Talking about the various field that we are having in a country to run successfully, we must say that hunger, poverty, sanity, food, everything are the basic essentials. Now, biotechnology has brought a lot of development in these areas. Talking about food, we can have so much knowledge where the agriculture has grown. There are increased yields in crop. There are increased, package materials, food materials. When we talk about pharmaceuticals, medicine, like antibiotics, so we are focusing on the health sector. It has immensely increased and it has immensely increased the population of the country because people are being more safe, people are being more healthy, people are having more lifespan, people are having more healthy lifespan. So in this sector, biotechnology has given us a very great help. Talking about the other factors, I must say sanity. We are having so many issues where sanity is a basic need right now, and biotechnology has brought us a lot of help in that sector as well. When we talk about healthcare and when we talk about food industry, we can say that biotechnology is not even helping us for sustainability, but it is also helping us to keep searching about the other results so that we can develop more of research. So there are a lot of people who are creating a lot of organizations. A lot of organizations are hiring a lot of people, so because of that, people are having a great help in economy wise. People are encouraging towards learning in these researches and applying for these for these researches, and that's why biotechnology has brought a great help for our developing country. Thank you.
So thank you, Samiran. Uh, our next participant is Janani Saptarshi. Is she there?
Janani, yes.
So you can start.
Okay. Hi, I'm Janani Saptarishi, and as the name itself suggests, biotechnology means it combines biology and modern technology for the human welfare. And I personally feel that biotechnology is not a very new field. Technology and development started right from when we can say Watson and Crick first discovered the structure of DNA. Till then, development has been in process, and and also through the early biotechnology, we have seen that farmers were able to select their best suited crops, having the highest yields to produce enough food to support the growing population. And even the curd which is made, even that has a very it's a very good example of technology being used in our daily life. And development of antibiotics, which is another very important use of biotechnology. Most of these methods involve selecting a final product with a better genetic makeup, and it is often used to the inclusion and this led to research and the field and establishment of biotechnology. And I feel that a country is intervened with healthcare, with agriculture, with sanitization, and all of these advancements can be brought up through biotechnology. And biotechnology has proved that it is more successful in treatment as compared to the conventional methods, because it works at a basic level of our body, that is the gene. And it has and it has also provide the permanent solution to decrease the dependence on medicines or injections. And through this we can even foster the integration for the latest developments in scientific research and all the engineering applications. And yes, and in such a situation, there is a constant need to improve the global health needs, and we have to put it in a effective we have to promote the effective public health measures, and development and advances for a country in such a field is a necessity, and more novel approaches through for therapeutics and low
Sorry to
cost
interrupt you,
treatment.
Janani. Please,
Yes.
can you wind up? Your time
Okay.
has already.
Okay, and
Oh,
that is why I feel that it has a varied range of applications for improving the variety and the development of country.
so thank you, Janani. The next participant is Ayushi Bhanushali. So, are you ready?
Yes, ma'am. So,
Okay, so you can
starting
start.
good afternoon, everyone. I would like to thank you for giving me an opportunity to speak. So, if you ask me the question, does biotechnology help in the development of a developing country? So, I would say it definitely helps a lot, because even if we consider the agricultural sector, it helps in the GDP raising in 16% approximately. So it is not necessary that the crops which are grown all give proper yield. Many crops are affected because of infertile soil, pests, everything. And biotechnology has helped a lot in this field, as using nanotechnology, we can improve the fertility of the soil. Also, pest resistant plants are grown. And I have read somewhere that banana is known to cure hepatitis B. So even if the researchers think something new and create a vaccine which can cure hepatitis B, it would help millions of people in our country itself and all over the world. Also, taking an example of maybe antibiotics, uh, biotechnology is immensely helping that, and we all know that right now in this situation, each and everyone is dependent on the biotechnologists for the vaccine of COVID-19. And so I would like to conclude by saying that the biotechnology wave of this year, or maybe from past 2 3 years, is similar to the information technology wave of 1980s and 1990s. Thank you.
Thank you, Ayushi. The next participant is Julie Kale.
Good afternoon, everyone. I'm sorry I cannot switch on my video. Uh,
It's
so,
okay, you carry on.
yes. So, does development of biotechnology help in development of overall countries? Yes, it does. Nowadays, biotechnology holds a tremendous promise for the developing world. The use of high yielding disease resistant crops would have a direct effect on improved food security, and it would also help us to get high yielding crops. Nowadays, biotechnology is being applied in ways that would affect the health, agriculture, environment, and many other science industries in a way which is unimaginable in the further decades to come. For developing countries, the promise of biotechnology is particularly very strong. Problems associated with population pressures, food insufficiency, and nutritional deficiencies, along with many environmental problems, would be solved. Uh, would be solved in a larger way. Also, it will also pave ways to new technological inventions and advancements. The increasing need for biotechnology is sorry developing countries and the governments of developing countries are also starting to understand the increasing need for biotechnology, and hence the governments of various countries have taken up many initiatives to boost biotechnology and to help the scientists and researchers. Thank you.
So, thank you, Julie. Now, the next participant is Priyanshi Kaushal.
Good afternoon, everyone. Am I audible?
Yes, you are audible.
Okay. So when
You
it
can
is
start.
asked, yes. So when it is being asked that biotechnology helps in overall development of the country, so I denied this statement. As for me, according to my views, for overall development of a country, the two main prominent sectors are of agriculture and the youth, which contribute in the overall development. So as we all know, getting a job in a biotechnology field is very hard. It is quite difficult for a fresher to get into the biotechnology field and get a job. Second of all, when the fresher is not getting a job after completing his bachelor's or BTech or MSc in biotechnology easily, then how it will contribute in the overall development of a nation. Second of all, coming to the agriculture sector. So when biotechnology is being talked about in relation to agriculture factor, it has some its disadvantages also. Like farmers are not very much well aware of the facts and about the techniques how to introduce them in agriculture practices. Second of all, it takes quite a good amount of money for them to introduce some techniques. And third of all, there are disadvantages as allergens and toxins, antibiotic resistance, potential of superweeds, escape. Let me give you an example to explain my statement better. So if you take a crop which is herbicide resistant, so it may transfer some of its trait to a weed, which would result in a herbicide resistant weed. So in that way, it is turning out to be a disadvantage. So these are two of the points which I strongly believe that biotechnology does not help in the overall development of a nation. Thank you.
Thank you, Priyanshi. Now, the next participant is Ayushi Sengupta. Are you here?
Yes, ma'am. Am I audible?
Yes, you are audible. You can start.
Okay. So, good afternoon, everyone. I'm Ayushi Sengupta, and today I'm going to speak about the topic that does biotechnology development help in the overall development of the country. So, to ensure an inclusive growth of a country, an innovation and metrics-driven approach needs to be adopted in various areas like agriculture, healthcare, science, and technology. So, here biotechnology plays a major role to ensure innovation and novel therapies. So, globally speaking, the rate of food production has dropped from 3% per annum in the 1970s to 1% per annum now. So, in increasing change in global climate is worsening the regional production of food crops. In developing countries like ours, agriculture is the main economic activity for a major chunk of the population who lives in who are living in the rural areas. So, without a successful production, they will neither have employment nor the resources they need for a better life. Also, many a times, a huge amount of crops is wasted to infection and infectious pests and viruses. So, biotechnology is working to produce disease and pest resistant crops, which can help curb this issue. It is also working on developing more nutritious strains of staple crops. Like most of us know about the golden rice. It is a modified strain of rice which packs more iron and beta carotenoid, which is a precursor of vitamin A. So, this innovation can help more than 100 million children who are suffering from vitamin A deficiency worldwide. Some researchers are on their way where fruits and vegetables can be given as vaccines one day. So, not just in agriculture, biotech plays a major role in medical advancements, pollution control, industrial developments, and many more. So, long story cut short, biotechnology aims to benefit society and it holds a tremendous promise for the developing world. Therefore, we cannot stress enough on the importance of biotechnology development in the overall development of a country. Thank you.
Thank you, Ayushi. Our next participant is Aditya Acharya.
Yeah. So yes, ma'am. I'll start. Yeah. So yeah. So one thing I would like to say here, as soon as this topic struck me, like as soon as you told this particular topic here, one thing I was very sure that, see, biotechnology seeks to generate new technologies using concepts and methods of modern biology that is there. But then one thing I personally feel that application of how biotechnology can help in the overall country is very specific. For example, application of biotechnology in a country like France would be completely different, and that too in a country like India, it's going to be completely different. So first of all, we need to understand that the application of this biotechnology is very specific. You can't, based on geographical location also, you see, for example, in France, it is already a well-developed country, so the application of biotechnology there would be different. But in here in India, we are still developing. So based on the topic, there were just two things which I struck upon related to India, and I would like to be very specific about India here, since that's our country. So first thing is, we are an agricultural based economy, so we need to pay attention to a lot of factors here. First of all, the current production is around 170 million tons of food grains per year, but based on the current population increase, the requirement would be projected around 250 million tons per year. Just see the gap in the gap and the increase, right? So we need to have more we need to have more increase, basically, I would say, in the agricultural based economy. So already people have said about how we can increase agriculture, agricultural production rate. So that is already going on through different type of biotechnology techniques, through plant tissue culture, etc., through genomics, basically, which is playing an excellent role. Now a very recent example of how we could have, prevented the possible effect of locust. A locust attack just happened, and it was like the most devastating one in a lot of years up to now. So you can see how biotechnology can act as a very good mode of action there. That agricultural biotechnology can actually play a role by not only developing and making more good crops, but also protecting our crops is equally important. Now moving ahead, one another very important thing is population. Our country is actually is suffering a population crisis, I would say, and which we are encountering from a long amount of time. A major problem for India and all other developing countries is the rate of our population growth. Increase in population to an extent is something to be proud of, but not to an extent that we are not able to control, right? So that is why India is currently 16% of the world's population, but just in a global area of just 2%. You can just see the difference. 16% people. There is also a map which just shows that in a round circle area of Southeast Asia, there are more people living in that particular area compared to the people living outside it. Can you just imagine? We are already sustaining so much, and there is still more to it, right? So a novel category of vaccine which I've just gone through, I guess, for controlling the fertility has been developed. One of these is actually under clinical trial in the first leg, I guess, of phase 2. So I personally feel that population is the, you just counter population, agriculture, everything comes under control. So population is the lead problem, I would say, and biotechnology is far more efficient to do that particular work. Thank you. Ma'am, you're muted.
Sorry, thank you, Aditya. So now I will call rest of the participants who were not there in the beginning. Yamini Purohit, is she here? Neha Bhandari, Srishti Pandey. Okay, then I don't think so. They have joined, and others have already talked. So thank you all the participants and our upcoming biotechnologists for such a nice discussion on this very important topic. So now I invite our guest speakers for their views on this topic and how well these students were able to present this topic today. So anyone, if they wish to give their views on them. I can see Jyotirmoy H ma'am, so if she wants
Yeah,
to.
yeah. A very good afternoon to one and all. And I think the speakers, especially our student speakers, they are just fabulous. I mean, the views they have put up, and the kind of perception they were having and everything are just superb. So I think some students are DY Patil students, and I'm always proud of my students, and that is the same thing that they have shown. I think one of the student like Priyashini, she has suggested something like it was little, but again, her point was also very true. We need to have amalgamation of biotechnology and the other fields as well, so that it is going to overall contribute to the development of India or any other country. And as per the Aditya has said, like from a country to other country it can vary, or Ayushi has said, I think all of you have your put up your views just perfectly. Thank you so much, guys. You are just really super. Thank you, people.
Thank you, ma'am. Thank you so much for your views. Now, can I request Dr. Praveen Podar, sir? I can see him also listening very
Yeah,
interestingly.
yeah, it was very interesting that the students, they have placed their views and in very short time, 2 minutes time, and it was excellent. Their idea is good, but certainly one student, I'm not remembering, who is talking about the opportunities and all these things, and that should be taken care by the government, because that is most needed. Most of the time the students, which I also experience, many students I also taught in my laboratory for their technological experience, that is lacking. So hands-on training, even in the colleges, they should have show given some small part of the research program to them, so that they can able to, when they graduate, they have some exposure to this. And I think that is very essential, which I want to tell these students to do that. And of course the organizers, they can able to put forward to the government that this such facility should be given to the colleges also. Otherwise, everything, the summit was very good for the students and everyone, and it was excellent. Thank you.
Thank you so much, sir. So, any other speaker wants to add on something? Richa ma'am,
Yeah,
Soumya ma'am, I can
yeah.
see you all.
So I'm also part of the placement committee of my institute. So many times where we face that there is a need of bridge between the education and the industry, and also so students, that part probably we need to develop focus more as a to develop industry ready professionals. One thing. Another thing is we need to create more job opportunities for the students, for the youngsters, because the number of students who graduate every year from all over India, different colleges, different parts of India, not everyone is able to get a proper job of their choice into the field. So those things probably from the organizer's point of view, if you can carry the message to the government that we need more investments. And for biotechnology, the problem is that investors has to wait for 5 to 7 years to get their returns. That is the main, I would not say problem, but that is biotechnology is. So as it deals with agriculture, it deals with healthcare sector. So probably more and more funding is required to sustain and grow to this biotechnology field. Otherwise, see, any sector, you name it, it requires biotechnology professionals. Any sector, any damn sector. So there's a huge vertical it has. So you can carry this message to governments that more funding, more opportunities, more job opportunities for the youngsters. Thank you.
Definitely, ma'am, we will try with all your help. So, any other, if they want to add on something?
I just want to say,
Yes, sir,
hello.
yes.
I just want to say like about the point of like funding from government for more opportunity for the work and something like that. I agree with that, but I'm first, I'm not Indian. I'm from Palestine, but I just want to say like Indian population is so high. So if you want to look for high population, it's not easy to get all of the effort from the government side. I think Indian they have like the choice to work in the startup, all of the private sector should prove it in this field, exactly in the medical field, because there is a lot of students. The population of India is so high. The number of the students is so high. So I don't think so. The government they will be able to support all of this and give opportunity for every student. I think also students they should start to work in that on their self and starting work in the field and starting working in the startup. This is what I think, and this is just opinion.
Thanks. This is a very nice opinion. So more number of startups which are social entrepreneurship and not only social entrepreneurship, rest of the things the government is already supporting us, but at the same time the policies need to be more streamlined so that startups will be able to sustain, not only function, they have to able to sustain and they have to be profitable. Otherwise, after 5 years, they will not be able to sustain. So those were streamline of policies is required.
Yes, ma'am, and yes, sir, you are very rightly said that students should take up startups, as our Prime Minister is also saying this only. So now, any student participant want to say anything? I am seeing one chat message. I don't know he's a student or a professional, but he says, I agree with Priyanshi that in world biotechnology is important nowadays, but it is also dangerous by some countries like China, who makes bioweapons too, which are too dangerous for the whole world. So he also said very correct. We should make use not in a negative way. We should be more productive towards it. So now, as the two-day National Biotech Summit came to an end, I would now proceed with vote of thanks. I, on behalf of National Summit 2020 and the entire organizing team of JAX Foundation, extend my hearty vote of thanks to all the dignitaries and our guest speakers present here. Our today's keynote speaker, Dr. Shilpi Sharma, Professor Savitribai Phule, Pune University. Thank you, ma'am, for joining us and enlightening us with your words of wisdom. All the eminent speakers for gracing their important work and sharing with us their findings and opinions. I would like to thank Dr. Lopamudra, ma'am, founder and president, Breast Cancer Hub, United States. She talked about challenges with breast cancer, connecting the developed and developing world. She told us about relation between arthritis and other inflammations of the body and breast cancer, which generally we ignore. Dr. Amit Kumar Pandey, sir, from Amity University, Haryana. He talked about non-coding RNAs as biomarkers in cancer, which is a very important upcoming topic of research. Dr. Pulak Mukherjee, Director, Institute of Bioresources and Sustainable Development, Manipur. He talked about drug from nature evidence-based validation, which is re-emerging in today's time more and more. Dr. Soumya Basu, Associate Professor at Cancer and Translational Research Laboratory, D.Y. Patel Biotechnology and Bioinformatics Institute, Pune. She talked about CAR T cell therapy, which is becoming very important therapy nowadays. Dr. Richa Mahant, Professor, Jivaji College, Gwalior. She talked about role of biotechnology in improvement of food quality and nutrition. Dr. Dhamendra Dubey, Assistant Professor, Symbiosis International University, Pune. He talked about understanding of data, biostatic approach. It is highly needed by each and every one of us in our studies today. Dr. Vijay Rajan Patnaik, scientist C. Ames, Delhi. He talked about breathomics, a perfect marriage between medical science and biotechnology. Sir, it was a very new topic and concept, especially for all student participants. Dr. K. Subramaniam, Professor, Banari Amman Institute of Technology, Tamil Nadu. He talked about development in biodegradable polymer biomaterials for biomedical application. I would like to thank our today's bright and young moderator, Mr. Aditya Acharya, Executive Member of JAX Foundation, for beautifully moderating the session. All the participants who were there for wonderful group discussion and all the lovely participants and academicians present to attend this. Without you all, it was nothing. Also, I would like to give hearty thanks to Director of Symbiosis School of Biological Sciences, Dr. Vinay Kumar Rail, who was a collaborator of this two-day national summit, to Mr. Jitendra Kumar, Director and founder of JAX Foundation, for perfectly managing his entire team, all the board of directors, Professor Kumadwala, JAX advisor, under whose guidance this foundation has started, Ms. Tanya Bhagat, HR coordinator and technical head, and the entire team coordinators who made this event successful. I would also like to inform all the speakers, delegates, and our participants that your e-certificates will be provided on your emails. All the students who submitted their papers and posters will receive participation certificates, and all those who are selected for final round, we will let you know by mail. We invite you all to be a part of JAX Foundation. You can surely drop us a mail and we will contact you. Mail ID is mentioned in chat box or they will mention shortly. Teasers, newsletters, and all the updates of two-day summit will be posted on social media. Thank you all once again for participating enthusiastically in the event. Take care and be safe. Thank you so much once again.